Good morning, everyone. Welcome to day 2 of the 2025 International Pharmacy Conference. My name is Henry Lee, and I'll be your MC for today. Before I begin, a few housekeeping reminders. Please keep your mobile phones on silent mode to avoid interruptions. In the case of emergencies, please follow the signs to the nearest exits. Refreshments will be served during the scheduled breaks, so please refrain from eating or drinking in the conference hall. Water is permitted. Lastly, for those applying for the continuing education credits, do remember to sign in and out. We do have a QR code outside the reception hall for you to access the sign-in form. Now, let us begin with the opening remarks. First, please welcome Professor Pei Shan Cai, Vice Principal of the Taipei Medical University, for her opening remarks. Let's give her a round of applause.
Good morning, everyone. So for the sake of time, I won't address every distinguished guest. So good morning once again. Welcome to the second day of the 2025 International Pharmacy Conference. Today, importantly, we will explore AI-driven drug discovery, advances in drug development, precision medicine, pharmacovigilance with insights from Japan, the UK, the US, and Taiwan as well. We have a great lineup of speakers today, trust me. You won't regret being here, all right? So I believe these sessions, I think, address very important issue, okay? That tell us, and you will find out from the speaker's presentation that, and you will be wondering, technology plays such an important role in transforming drug discovery, precision medicine. Not only that, I think our session reminds us the importance of patient safety. We need to keep that in mind, all right? And I believe you will learn a lot from the session. But I strongly encourage you not only to learn from our experts, but also to engage with our experts, connect with each other, and hopefully build connection and collaborations for future perspectives. Once again, welcome to the International Conference of Pharmacy, right? And I wish you a productive day ahead. Thank you.
Thank you, Vice Principal Tai, for your inspiring remarks. Next, we are pleased to invite Jingshun Huang, President of the Federation of Taiwan Pharmacists Association. Let's give him a round of applause.
Da家早安。蔡副校长、韦乔院长，还有来自美国、日本、英国各国的药学专家，还有今天关心我们药学发展的所有好朋友，大家好，大家早安。非常荣幸，今天代表药师公会全国联合会出席2025国际药师研讨会。 健康照护与制药所学领域的次世代药学。首先，我要感谢主办单位与台北医学大学用心的策划规划，让我们可以聚在一起，共同讨论台湾的药学教育、临床服务与研究创新的未来方向。这场会议的主题。 我认为特别具有时代的意义。药师的角色正在全球医疗体系中快速的发展。过去，我们被视为药物的供应者，但今天，药师更是 病人用药安全的守护者，精准医疗的协作者，公共健康的推动者。这个转变不仅发生在台湾，更是在国际上共同的潮流。在台湾，我们同样面临许多挑战与契机。随着迈入超高龄社会，慢性疾病与多重用药的情况越来越普遍，药师必须投入更多的药物治疗管理，协助病人提升生活品质。同时，人工智慧与药物、基因、体学等科学的发展，正逐步改变药物研发与临床决策的模式。药师也需要持续学习，与时俱进。而在用药安全与制度的改革过程中，药师更肩负起第一把关责任，确保专业制度不断精进，守护全民健康。这些课题还有一个国家没有一个国家可以单打独斗，因此国际交流显得格外的重要。我相信，随着透过这次研讨会，我们可以彼此学习，互相启发，从药学教育的革新到临床药学与药物遗传的应用，乃至 AI 推动的药物研究与安全监视，每一个议题都关乎下一代药师该如何成长，药学专业如何精进进步。作为药师公会全国联合会理事长，我特别透过这场会议，能让台湾的药师专业领域国际紧密结合，唯有合作，我们才能面对全球性的医疗挑战，找到更好的解决方案，并让药师的专业价值能更广的认同跟发挥。最后，祝福今天的研讨会圆满成功，谢谢大家，谢谢。
Thank you, President Huang. Thank you, President Huang, for your encouraging words. Finally, we are honored to welcome Yu Li Zhang, President of the Taiwan Society of the Health Systems Pharmacists. Let's give him a round of applause.
蔡副校长、赵博士、张院长 our distinguished guests, ladies and gentlemen, on behalf of Taiwan Society of Health System Pharmacists, it is a great honor to co-host this conference, and it's true pleasure to welcome all of you to second day of the 2025 International Pharmacy Conference. First, I'd like to express my sincere gratitude to Dr. Alan Zhao for his invaluable support and contribution, and also to the School of Pharmacy at Taipei Medical University for organizing such a high-level conference featuring visionary topics that both broad in scope and rich in depth. After yesterday's full-day inspiring session, I am sure many of us are eager for more and look forward with great anticipation to today's program. Today's session will focus on AI-driven drug discovery and development, as well as precision medicine and pharmacovigilance. In the morning, we will gain new perspective on statistic genetics, synthetic biology and AI-based prediction of immunotherapy responses. In the afternoon, we will turn to the application of genomic information in cancer treatment and the challenges of drug safety and the risk management. With such distinguished scholars and practitioners, the discussion promised to be both stimulating and rewarding. I'm also pleased to share exciting news for you. To promote precision medicine, our society, TSHP, collaborates with Dean Professor Weiqiao Zhang and the Taiwan Pharmacogenomic Society. Together, we will publish in October a new book entitled Clinical Service Guideline for Pharmacogenomic Application. This practical guide is designed to support physicians, pharmacists, and healthcare professionals, ensuring that the ideals of precision medicine can be effectively implemented in patient care. I firmly believe that through cross-team, cross-country, and cross-disciplinary collaboration, we can achieve even greater breakthroughs in both clinical practice and scientific research. Finally, I wish this conference great success and hope that every one of you will find today's program both fruitful and inspiring. Thank you, and I wish all you a productive and rewarding day.
Thank you, President Chiang, for your thoughtful remarks. Now we shall move on to our group photo. We will have our designated photographer to take a group photo of all of us. So please, bring out your best smiles. Yeah, if those who have your masks, please take them off. Thank you, everyone. Now, let's officially begin the conference. To moderate our first presentation today, we're pleased to invite Professor Weiqiao Zhang, Dean of the College of Pharmacy, to introduce our first speaker. Let's give him a very warm welcome.
Good morning, ladies and gentlemen. It's my great honor to introduce my longtime friend, Professor Yukinori Okada, who I regard as the best, the most outstanding physician scientist, now a professor at University of Tokyo. I first met about 20 years ago when we were both working in Riken in Japan. And even back then, he was already a superstar. After graduation from University of Tokyo, Department of Medicine, he completed his postdoc research at Broad Institute, MIT, where it's the best genome medicine institute. And with very short time, he became a professor in Japan. I believe Professor Okada is the youngest full professor in Japan. And as far as I know, he published more than 30 papers in Nature. And I still remember that many, many years ago, when most people were not familiar with polygenic risk, or PRS, he was already a pioneer in this field. In 2013, he published a landmark paper in Nature that described how to use GWAS data for drug discovery against rheumatoid arthritis. A paper that has already been cited more than 3,000 times. And a year later, based on his concept, I worked with him to publish a paper focused on colorectal cancer. And following his concept, following his leadership, we have all learned dramatics from his vision and innovation. Professor Okada is incredibly busy, and this time he is only staying in Taiwan for less than 24 hours. We are truly honored to have Professor Okada. Let's welcome Professor Okada.
Okay, hello everyone, this is Yukinori Okada from the University of Tokyo, Japan. And again, thank you so much for kindly inviting me, Wei Chao, and have a talk at this wonderful symposium. So today I want to talk about not fully solely on drug discovery, but more statistical genetics, how they can contribute biology, medicine, drug discovery. But okay, but before starting talking my talk, I think the most way to show respect to speed learning. So maybe I should show my training of Google Translation. Okay, okay. 台湾的朋友大家好，我的名字是冈田随尚，我是日本东京大学的教授，我很高兴见到你们。Thank you. Okay, this is the only Chinese which I can speak, so I hope you guys understand. Okay, let's start the talk. So my specialty is genetics. So we're always thinking of having genome sequence. So we are the genome sequence is the blueprint we use. If you can fully understand the meaning of this sequence, you can reconstruct yourself. Also, but we are still at the tip of the iceberg. We'll definitely go on to the bottom of the iceberg. So how we can extract biology or more knowledge from genome sequence? This is a very important point for us. And I have been working on this genetics, disease genetics stuff for the last 15 years. You may know the name of GBAS, genome association study. This is one of the most successful methodology in the field of statistical genetics, which identifies so many de-risk genes. I think there's more than 1,000 genes for 1,000 traits. It means that if you're a medical physician, you mean that most of the disease which you learn to pass the examination of physician, most of these already has some genetic studies. So there's so many wide achievements. And yes, I have been working on the Biobank Japan project, which initially launched from Professor Yusuke Nakamura more than 20 years ago. I think it's now one of the initially launched disease-based biobank in East Asia, and also it has been a very successful one. So we have been working on it, and now I'm working with Professor Koichi Matsuda on this. Anyway, so many 260,000 individuals, and we have collaborating many, and we providing so many results so that anyone in this field can utilize it. But anyway, we are very open for collaboration. If you have any interest, please let us know. And previously, we conducted genetic study, each paper for each disease separately. But now, because Biobank has so many information, we are now massively doing such analysis in parallel. For example, this is the achievement from Saori Sakao and Masahiro Kanai. They're both working at Broad Institute now. And yes, but we conducted GBAS more than 200 phenotypes along with UK Biobank, FinGen. And yes, we found so many disease-associated variants. And what is most important in this field is make the result publicly available. So we constructed a database named fieb.jp. So please go to this website and and get your result. And also I'd like to talk on something related to some specific disease. So this is pan-cancer GBAS result. Previously we conducted GBAS separately for each diseases, but for example, this is an in parallel conduction of GBAS for multiple cancers, and we found several variants which are shared across the cancers. Or we find some shared genetic background, especially between prostate cancers and breast cancers. So these two cancers are rarely occur because of the difference with the sexes, but these two factors strongly share genetic backgrounds. And maybe project such a GBAS result into single cell references, we can pinpoint some causal epithelial cells, which has a strongly responsible roles for the development of these two cancers. So anyway, GBAS staffs now provide so many results, including implication of these risk genes. And again, this is our website, so fieb.jp. So let's go to this site, and please input your name of disease or gene or genetic variants of your interest. You can get the visual result, and also you can download the results from here. So this is something result really in type 2 diabetes. Yeah, I think I want to ask the researchers who is not specialist for genetics, non-genetic researchers, I would like to ask to have interest in such results, because these are very easy to provide you more information related to your field researches. Okay, so, but I have been working on GBAS, but now GBAS itself is now shifting to, it's no longer pure science. So 20 years before, GBAS was very pure science, right? But now it is changing more infrastructure. So pave the way to so everyone go further. So open and everyone can access. This is a very important point of GBAS. So maybe we should be keep doing these efforts to further provide the results to the community. And also our biobank itself need to go forward the new challenges. So this is a new challenge for Biobank Japan. So Biobank Atlas Japan project, we are now conducting brain multiome atlas. So we found that the similar overlap of Biobank Japan and brain biobanks. Now we are massively doing single cell or short or long read RNA sequence and genome sequence with these brain sample so that we can get the new result. Because brain disease is very important for the aging days. Okay, so this is the introductory talk. So today I'd like to tell you several omnibus talk which we have been recently have interest. The first point is, yes, germline and somatic mutation, how they both interact in getting some diseases. Some of you are cancer guys, some of you are non-cancer guys, but I think both cancer and non-cancer guys should have interest in both two factors. So, okay, this is, I need to talk on first of the some specific region in the genome. So this is the GBAS result of rheumatoid arthritis, which is one of the famous autoimmune diseases. And you find that there's a strong peak at chromosome 6. So this is well-known HLA gene and MH region. So this region and chromosome 6 has a strong impact on your acquired innate immunity of yours. So we already see strong peak at this peak when we do GBAS of immune-related phenotypes. Okay, it makes sense, right? And I'd like to need to some detail because I have been working with this region. So here's HA region, MSC region. They include multiple HA genes, and they have so many tons of HA variants. You know, this is one of the most polymorphic sites in the human genomes. And we conducted the HA imputation to staff. So getting computationally impute HA arrays from the surrounding shapes, which initially developed by Shomore Chaudhuri at Harvard, and now we apply this to Japanese population, yes, which introduced this to the Taiwanese population. So anyway, we can now know which disease which HG has the risk. And for example, this is the result in the rheumatoid arthritis. So HLA-DLBM molecule and some amino acid polymorphism as the specific critical region of this molecule, they have strong risk of, for example, seropositive RA or seronegative RA. Also, some non-classical HRA also affects them as well. But anyway, HRA imputation and fine mapping, especially amino acid sequence position, is very critical to define the risk of such immune-related diseases. So this is the association with the HLA variants, the phenotypes, but recent development of single cell provides us how they affect the TCRs. So recently we conducted the single cell EQTL database of Japanese, and you see that these HLA alleles and TCR co-interact together and getting to bind. So there's some connection or restriction with HLA variants to which TCR to utilize. So, and of course, class 1 HLA is associated CD4 T cell TCR and class 2 for CD8 T cells. And but we found that HA class 1 restriction to the TCR is very specific when we are doing the interaction with COVID-19. So it means that under the disease status, how HA variants regulate TCR changes. So we definitely need to see this interaction each disease separately. So this makes sense. Yes, HA variant is strongly important for your disease or TCRs and anyway. But I have been thinking of one question. So because these are very critical region here, so what happened if this region go away from our body? So is this the immunity goes well or bad, or it's a long question because cancer guys say that in the cancer cells which lacks MHC molecule evade from immunity and they are getting worse cancer. So I think perhaps if we partially lacks this region from our body, it should be something wrong. So this has been my long idea. And then we get back to these findings when we conducted the genetic GWAS of COVID-19 vaccination titers. So the University of the Keio University in Japan, they constructed a cohort of genetics and COVID-19 vaccination antibody titers, and we conducted GWAS, and we of course found many genetic variants associated with COVID-19 vaccine titers, but finally we find that somatic mutations strongly regulate such antibody titers. Okay, let me introduce this. So firstly, we conducted GWAS for difference of the COVID-19 vaccination antibody titers. And yes, we found strong peak at the MHC region at chromosome 6p, which really makes sense. And we also found another genetic variant at chromosome 14q. So it's a immunoglobulin locus, constant region of immunoglobulin. Here's a East Asian or Japanese specific missense variant, which is located in the immunoglobulin receptor binding site of the constant region. And people who have this missense variants are likely to have lowered vaccine titers. And also the influenza vaccination titers also lowered. It means that the genetic variant is strongly diminishes acquired immunity. So germline variants chromosome 6p and 14q both strongly affect the acquired immunity difference. So it's very interesting, but it's a kind of expected result, right? Expected result when we always do GWAS. But then we focusing on somatic mutation. Somatic mutation, especially called clonal hematopoiesis or mosaic chromosome alteration. So clonal hematopoiesis is a kind of age-related somatic mutation accumulation with abnormal clonal expansion. I don't know why, but some cells which have somatic mutation are starting to quickly increase, clonally expand. And there are two types of clonal hematopoiesis, mosaic chromosome alteration and also gene level TET3 DNA MT3 mutation. But anyway, many people now have interest in how these clonal hematopoiesis and somatic mutation affect human disease risks. And then Kyuto Sonehara, he is now working at the Sanger Institute. So he conducted this somatic mutation association study with COVID-19 antibody titer using the public UK biobank data. And we find that somatic mutations chromosome 6p and 40q, they have a strong impact on COVID-19 vaccination titers. Again, this 6p and 14q, this is the region which includes MHC region and IGH region. So the two hits in the GWAS. So hits in the germline GWAS and hits in somatic mutation was the same. This is a very interesting result, right? So when we focused the somatic mutation into MHC region and the IGH region, their association gets stronger, especially people who have somatic mutations MHC. They're no seropositive guys. They couldn't make antibody. So these two somatic mutations have strong impact on acquired immunity. Then we checked into the association with disease. Of course, they increase the risk of infectious diseases, and they also increase the risk of immunity, especially Graves' disease. I don't know why Graves' disease risk increased, but in both UK Biobank Japan, somatic mutations, MHC or IGH increased risk of Graves' disease. Anyway, these are not good for infection disease, but also the immunity diseases. And then we will also from sex chromosome somatic mutation. sex chromosome is more likely to be easy to get lost, and loss of Y and loss of X, and we found that loss of Y strongly decreased the COVID-19 vaccine seropositivity. So we know that the aged male are vulnerable to the COVID-19 infection and less likely to be fully vaccinated. One potential reason is loss of Y effect. And of course, loss of Y and loss of X increase the risk of infectious disease and also increase the risk of asthma or rheumatoid arthritis autoimmune disease. So somatic mutation in some specific region of the genome, they increase the risk of, they diminish acquired immunity, but also increase the risk of infection and immune disease. So it means that when we want to thinking, especially the aged guy, we definitely need to consider both germline and somatic mutations. Very nice, very nice. But I agree, discussions on somatic mutation risk is still controversial. So here's a very famous paper in Nature Medicine. Chromium hematopoiesis is protective for Alzheimer's disease. But of course, I know this paper, what the analysis is right. But if any of you want to increase your somatic mutation to prevent Alzheimer's disease, please raise your hand. And I think no one raised it. This is because a little bit biologically confused. So still there is discussion on causes and consequences of somatic mutation. This is because we don't know what they are doing at cellular level. So to understand these causes and consequences, we should definitely understand the cellular states of how somatic mutation is doing. Okay, let's go on to how to project such somatic mutation into single cell. So when we project single cell somatic mutation into single cell, we can now understand what they're doing. So we definitely need new technology always provides us the new science. So one of the not completely new but relatively new technology single cell sequence. You see many this kind of figure, bulk RNA sequencing, single cell sequence, something like smoothie and before smoothie, right? Anyway, it's a kind of advanced technology which tells you the cell type specific resolution. So we just started this technology during COVID-19 pandemic. So initially we started to collect so many COVID-19 patients and conduct single cell sequence. Recently, here comes a very nice technology to project such population scale data, something like GBAS result, into single cell resolution. For example, this is a projection of COVID-19 GBAS result into single cell stuff, and cell which expression changes, the disease risk gene, the expression changes, such cells are going to be identified. In this case, we found that severe COVID-19 project risk is strongly eliciting innate immune cells. So we can know which cell is very causal for such phenotypes. And motivated by these findings, we then expanded our resources to Osaka Atlas Immune Cells, OASIS. In my laboratory, the people who developed the resource have the naming rights of the database. So, okay, so he named OASIS. Anyway, we collect COVID-19 patient, health and conduct single cell sequencing. And yeah, once we have such data, we can do so many types of analysis, something like EQTL, a single cell EQTL analysis, or dynamic EQTL, cell type specific, T cell, B cell, but the main focus of this study is coronary hematosis. Okay, we now have single cell. How to project somatic mutation into single cell resolution? So this is why we try to do it. So the idea is something like this. We can know who has the somatic mutation in which point of the genome used based on the population scale data, right? We can know it. And then using such somatic mutation as a prior information, we project them into the single cell. And then we can know which cell has somatic mutation or which cell do not have. So without human genome, it's very difficult to distinguish because single cell data is still noisy. But using this population scale data as a prior, we can understand which cell has risk or somatic mutation or not. And any biology is finally cell type specific. So if you can correctly project such somatic mutation, if it's correct, we can finally get the cell type specific result. And this strongly worked well. So this is the result. What is fortunate is COVID-19 patients. COVID-19 patients are very enriched with coronary hematopoiesis. So because coronary hematopoiesis itself is a very rare event, but we could get multiple patients. For example, this patient has two somatic mutations, chromosome 1p loss and chromosome 15q gain. And you can see that in these people, somatic mutation is only accumulated in here, monocyte in dendritic cell. If you put project such somatic mutation into UMAP single cell, there's only here somatic mutation, right? So somatic mutation accumulating here. Okay, within this monocyte clusters, why not compare the cells with somatic mutation and cells without somatic mutation? And of course, we found many dysregulated expression, especially chronic inflammation status. So once cell has mutation in monocyte in such chromosome position, they are getting chronic inflammation. This is one of the reasons why coronary hematopoiesis has the disease risk with chronic inflammation. Anyway, very successful, right? They're enriched. And this sample, we couldn't find the strong enrichment. But you find that not everyone has the same cell type specificity. These people, 17q gets, he has an accumulation B cells. Okay, why not check B cells? We checked B cells. And yes, in these people, somatic mutation is only at B cells. And because they're coronary expanded, this cell has only one TCR, BCRs. So we reconstruct BCR and found that these BCR recognize nothing. So these people are coronary expanded B cell, keep making BCR which are very meaningless BCR. This makes him vulnerable to the infectious diseases. Oh, so when somatic mutation has different position, different structure, there comes a different story. Okay, let's go to the sex chromosomes. So we then check the loss of Y. So loss of Y is always very simple, right? Very easy to calculate each cell. Which cell lacks Y chromosome, which cell do not lacks. And you find that loss of Y is mostly accumulated in innate immune cells and monocyte dendritic cell. But people who has COVID-19 specifically losing their Y chromosome in dendritic cells. I don't think they start losing Y after COVID-19 infection. So they originally loss of Y, and yes, they are risk for COVID-19. And when the monocytes are going to lose Y chromosome, and their T cell regulation pathway is starting to change in these monocytes, and then finally, this changes the composition of T cells. So when the people starting to have loss of Y, such monocytes in this cell corona is starting to expand, but T cells are going to be, so they somehow differential abundance of upper myeloid cell, lower naive cell. Anyway, there's some dysfunction in T cell composition changes. So here is another story for chromosome Y. Then we try to, as much as possible, which type of somatic mutation we can project. So we then focus on mitochondrial heteroplasmy. So mitochondrial heteroplasmy is a somatic mutation, mitochondrial sequences. And yes, it's very easy to calculate mitochondrial somatic mutation. Here's the mitochondria. And we found that this, and mitochondrial gene is also expressed single cell. We always discuss them after QC, but they're very informative. So when we check these missense mitochondrial mutations, yeah, they are strongly enriched in monocyte and dendritic cells. So they're also specific. And here's a fraction of cells between whole genome sequence. It means whole blood and PBMC. You can see that if you have some one specific function of mitochondrial heteroplasmy, they change the fraction. It means that they have some effect on cell proliferation, separate for PBMC and granulocytes. Anyway, mitochondrial heteroplasmy itself is very cell type specific. And but when we talk this, people always say, how about T cell, right? T cell is most important cell. How about T cell? So we look for coronary hematopoiesis disease risk on T cell, and occasionally found going to the neuromyelitis optica, NMOC. It's a kind of the new disease. There was no NMOC when I was a medical student. It's a kind of autoimmune positive type of multiple sclerosis, but now the aquaporin antibodies. So we have the chance to get this rare disease sample only 200, but sometimes larger number. And when we conduct GBAS, we found some HLA and CCL, very immune disease-related phenotypes. So it makes sense, right? So it's very kind of expected result. But when we conducted coronary heart association, we found that coronary hematopoiesis has a strong risk on NMOSD. Not so for other immune diseases, but of course they are famous for blood cancer risk, but strong risk NMOSD. So we then started to correct the PBMC of the NMOSD patients who have somatic mutations, and they are mostly accumulating T cells. So coronary hematopoiesis NMOSD is characterized by 21q loss, and they are accumulating CD40. And 21q have many interferon-related genes. So if CD4 T cells starting lose or increase these chromosomes, strongly they have got dysregulation of interferon, right? So there's so many stories how coronary hematopoiesis is a very wide heterogeneous. Which point of the chromosome and which cell type, these two combinations defines the immunology. Otherwise, we definitely need to make the catalog what they are doing. Okay, this is somatic mutation, but I'm going to talk a little bit different part of the association for disease risk. So virus risk for SLE. So here's a long-standing mystery of virus infection. So the link from virus infection to and onset of autoimmune disease, it has a long history. So this is a paper written 30 years before my mentor professor Kazuhiko Yamamoto. It shows, yes, here's a similar amino acid sequence, herpes simplex virus, and a RNP antigen. So people who are infected with herpes are likely to get antibody disrecognized, and it causes autoimmune. It's a kind of, mystery, right? It makes sense, but it's a kind of still speculations. But after 30 years, we now have more advanced technologies. So here comes the COVID-19, right? COVID-19 pandemic, we again experience the importance of the understanding of virus infection. So it's a very tough time for us, but this provides us a new networking of researchers. We construct Japan COVID-19 task force led by Keio University. They collected more than DNA, RNA, clinical information set of more than 6,000 COVID-19 patients. This is very impressive because we cannot get new COVID-19 patients because COVID-19 itself changes. And during some GBAS, we find some critical gene or some. Anyway, we found that virus infection is a strongly important factor for our health. So after collecting these sample and doing analysis, we then focus starting the virus-oriented disease analysis. So non-human reads. So we conducted whole genome sequence of human blood. They provide so many human genome information. But human whole genome sequence includes shallow non-human reads. Non-human reads, most of the case, comes from virus. So we get the non-human reads and maps them into the virus information and quantified the endogenous and exogenous virus infection status. Endogenous virus is a rare type of virus which are genome integrated with germline human genome. It always happened for human herpes virus. We finally found they have the risk of autoimmune diseases. Okay, so we focus on human herpes virus 6B and 6A. They are kind of well-known endogenous virus which go integrated in the human genome. And when we evaluated the frequency of this EH6A and 6B across control, in 6B we found strong enrichment of this herpes virus in rare type of autoimmune disease, SLE, systemic lupus erythematosus, and pulmonary alveolar proteinosis or multiple sclerosis. And then we checked the disease activity. SLE patients who are positive for endogenous herpes virus 6B, they have strongly increased activity of the disease. So they have a very severe type of SLE. So there should be something wrong for immunity. Then we get the corrected PBMC and conducted SLE, and we found upregulated monocyte signals, interferon signals in monocyte. So anyway, this EHGB6B positivity causing something wrong and causing the severe type of herpes viruses, SLE disease status. And we also checked whether they are truly integrated to human genome or not. So we conducted GBAS of around 20 EHGB6B positive patients and negative one. And again, we confirmed the peak at chromosome 22q longer. So human telomeric sequence is very similar to those around human herpes virus. So telomeric region is the site of the human EHGB integrated into telomeric region. So we found that. Yeah. But it's interesting, different population, different integration site. I don't know why, but this rare event occurred multiple times in human history, and they should have some positive aspect. This is because why these genome has been keep segregated. Okay, there should be something wrong in EHGB6B, but we further checked the immune phenotypes. So we conducted, or our colleagues and we can conduct the FIP6. So FIP6 putting the virus sequence into phage display and put the antibody and check the activity. So here's the sequence of human herpes virus. We found that people who have EHGB6B, their antibodies recognize very specific position of the herpes virus. Other people, of course, they are affected with herpes virus. They are using some different products, but anyway, I don't know this is a critical region or protective or pathogenic or antibody, I don't know, but it means that the people who have AH6B from Z1, they have very specific immune profiles. And also we parallelly checked many virus affection status, which is exogenous virus, and we finally found that anaerovirus loads. So we quantified the anaerovirus load using the non-human raising human whole genome sequence data. And actually, we quantified their curves, and you find that rheumatoid arthritis, SLE, COVID, they have increased anaerovirus loads when compared to controls. So anaerovirus has been reported many COVID-19 patients in the ICU unit. Yes, we found that they reflect the many infection, virus infection statuses. Okay, so we now know that somatic mutation and virus infection strongly affect the status of our disease. And human disease, strongly human genetics helps us to understand. So finally, I would like to talk on some genomics-driven drug discovery. I myself is not a pharmaceutical guy, but we somehow do something related to drug discovery. You may know that this is a very famous barrier. So to get finally launched the drugs into society, we need many phase 1 or many targets, and day by day it became more severe and more highly costing. And so we consider around 10 years before, okay, here's a disease, here's genomes and drugs. So previously we tried to understand drug from disease biology or from genome to understand disease biology, but human genetics is very strongly related to the disease risk gene, so why not directly check drugs from human genome? So genome-driven drug discovery is a direct way from genome and drug. Sometimes we do not understand disease biology. They help us to understand the connections. And after 10 years, this makes common sense. So this is a very famous review in Nature Review Drug Discovery. Okay, among the 50 drugs approved by FDA in 2021, 2/3 of the drugs had support from disease genotype phenotype association. So human genetics reported the links between phenotypes and disease risk of the gene of the target of the drugs. And the largest population comes from common diseases. It means GBAS, right? So GBAS is now the most promising resource for drug discovery. And then our laboratory has been working on how to connect human genetic resource with such drug resources or biomarkers. For example, MyGBAS, this is a microRNA screening with tissue-specific microRNA. So when we put GBAS result, tissue-specific microRNA expression profile and their target network, they provide us the list of the target gene and biomarkers. And here's the graphs. So when we put the GBAS result, expression data, cancer somatic mutation, any kind of the gene list which was screened in hypothesis-free, when we put, I'm going to provide a list of the candidate drugs. Maybe I show some more details on this GRAPE scheme. It's a kind of automated network of GBUS target drug disconnection. Here's a GBUS result, and here's a target gene, the compound, and here's the compound, and here's a disease risk. So when we connect these layers unbiased way, when you put the left one, you can get something right one. So here's a GBUS connect here, and some database connected drug compound target, and here, for instance, WHO ATC classification provides a list of the current disease and currently used drugs. So this is a very simple scheme. This software is very simple, but when you put disease risk, this automatically calculates which disease is more promising for drug repositioning, which compound is promising for drug repositioning. So yes, this scheme somehow makes well, right? But you found that we are not considering any directions. So this is one of my old paper, more than 20 years. So this shows the overlap of drugs used for treatment RA and RA risk gene of drugs. They have strong connection, but you may know that we are not considering any direction, just overlapping, right? Whether we should up or down the gene, we are not considering. But if you consider drug development, you should definitely understand this direction. So I want to try to shift more different. In strict drug study with gene expression dynamics. So this is the idea. So each circle represents genes, and x-axis means gene expression changes when you put compounds, and y-axis changes when you compound changes. Case control difference between expressions. So if they have negative correlation, this compound may have the potential to ameliorate the disease status. So we expand from simple overlap to gene expression dynamics. And there are many databases for rights, but here's case control difference of gene expression profile, especially in tissue-specific ways, is still very difficult to get because, yeah, so difficult to collect so many patients and tissues. So we utilize GBUS result. So when we put GBUS result, and when we integrate them into EQT result, it means that how genetic barriers change the expression profile. So we can understand the case control, simulate expression change difference with disease status in tissue-specific way. So we constructed a software named Transfer. Yes, Transfer can identify drugs implicated for input GBUS. So when you put GBUS results, Transfer calculate EQTL and calculate the case control expression difference changes, and they're going to find a negative correlation with the compound perturbations. And still that this expression estimation is still some noisy, but they somehow do work. So when we develop this software, here's the COVID-19 panic. We put severe COVID-19 GBUS result. They found verapamil with hematopoietic cell should be protected. And I found there was some clinical trials on verapamil COVID-19. So if this clinical trial goes well, I plan to say I found this drug. But regrettably, there was no further news for this. So there is itself changes. But anyway, consideration of disease expression change dynamics is very critical when we apply human disease genetics into further development. Okay, then several years later, we proposed the best practice of genomics-driven drug discovery. So here's the GBUS result, or GBUS result. It is going to accumulate automatically within society. There exist multiple ways. So direct disease gene, just looking simple overlap, or some QTL Mendelian randomization. I don't talk detail here, but yes, case conjugate expression changes by theory. This is what we talk. Anyway, there are multiple ways to how to utilize automatically increasing human disease genetics, and yes, they provide a somehow rational list of how drugs are doing well. And yes, this is but then the result of how we apply GBUS, and here's a gene. Anyway, I see this is some example in gout or PT, or anyway, if you put the detailed GBUS results, which most of the case publicly available, you can pinpoint which category of the disease for multiple ways of the method. Yeah, if you have any interest, please just show. So now I mean that GBUS result, chemistry-driven compound screening is now a very common way to in this field. And then the collaboration with the GBUS on stroke guys, we applied our method. And here's several methods. When we put GBUS result of stroke, they pointed at several compounds. Some has already been approved. Some is now under active discussion, and some is anyway. This method provides a very relatively effective list of the compounds. And what I'm now currently doing is applying our compound list to more in vivo screening. So people who have patient-derived iPS cell, I ask them to put my compound and going to check how they changes. Or if the mouse model, how they change. Anyway, in silico screening itself don't change the society, right? Combination of in silico screening and collaborative in vivo screening is very important to proceed for a drug discovery forward. Oops. Okay, this is almost the end of my talks, but anyway, I think developing young scientists is very important. So every summer, I have 2 or 3 day course of statistical genetics, which I just finished. Yeah, several days before. So it's I have already opened the 10 times of this workshop. But anyway, development such statistical genetics, computational knowledge is very important. So we also introduced this computational code by this course as the Osaka University of Tokyo. I think this is very important to be research in any fields of the biosciences. Okay, I would like to say thank you so many collaborators, especially to excellent laboratory member Ryuya Edahiro and Gosato, who leads the single-cell academic studies. I would like to say thank you. And Biobank Atlas Japan is one of the most active research fields in here. Maybe 1 or 2 years later, I would like to introduce how they are going. Okay, thank you for listening.
Wonderful talk. Now we are open for discussion. Any questions from audience? Okay, Dr. Wu.
I hope I say it right. 勉強になりました。And a wonderful talk and a very excellent achievement. So actually, as a physician and like treatment medicine, so my first question is that I'm very interested in the mosaic chromosome alteration events in topic 2. So I'm wondering, maybe you already mentioned that. So will that impact the outcome of those infection, and will that like in your topic 4, will that guide the clinical decision in treatment or?
Okay, but thank you for, it's a very important point, right? So, okay, I admit that the risk of mosaic chromosome alteration is still debate. The one point, as you said, most of the association is just seeing retrospectively. We should definitely check prospectively to how the basic status of the coronary hematopoiesis risk of onset of disease. But you know, most of the coronary hematopoiesis events are very rare. And disease onset is also rare. So actually, most of the case we don't know this. So this is the reason why there is multiple controversial discussions the risk of coronary hematopoiesis of Alzheimer's diseases. And the other aspect is I thought that we don't know the biology, right? We should definitely classify coronary hematopoiesis based on the biology, especially combination with serotype specificity and chromosome positions. So there's no catalog. So it's a kind of still still interesting, but not yet enough to be informative to put them into the decision making.
Thank you.
Okay.
So thanks for the fascinating talk. So quite interesting about your first part of the somatic mutation caused by the coronary hematopoiesis. So it seems like it's not only the SMV mutation, it's also including the structure variation. So do you know what's the drivers to cause this somatic mutation or structure variation? Is this the virus infection cause it? And how did you ever comparing the samples before and after infections? And my another question is that this kind of the somatic mutation, are they do they have like the virus specific signatures? So this is my question. Thank you.
Very important points, but my quick answer is we don't know yet. So. Yes, multiple types of coronary hematopoiesis, chromosomal events or point mutation. But we know these two types of mutation likely to occur together. So, but we can observe coronary hematopoiesis, but we don't know why chromosomal hematopoiesis. What is the driving force of cell expansion? Right? This is we don't know why. We don't know why they are likely to expand. So we don't know. And the other question is when they initiated. So we can observe somatic mutation along with aging, but it doesn't directly mean they initiated after aging. Some somatic mutation can occur much earlier stage of the ages, just because we cannot observe them. But anyway, we don't know. We still not know why they expand or when they initiated. And of course, we don't know what they are doing biologically. I think hematological cancer researchers are massively doing this. And some somatic mutation or there's a model mice, something DNMT3A point mutation mice or loss of Y mice, they exist. Maybe some biological aspects can be assessed in such using some mouse models. But anyway, most of the cases, they are completely mystery still yet.
Any questions? Okada-sensei, when we evaluate the quality of messenger RNA vaccine, usually based on the antibody titer. If the antibody titer goes high, it might be judged as a good responder. And my question is, does any variant or any TCR that is able to classify the booster vaccine antibody titers?
We haven't get yet. So what observed is Jamurant, MHC, and IgH. IgH may regulate BCR, B-cell maturation, and HLA variants which affect IgG titer may interact with some specific TCR, right? So, but we haven't get any result yet. But maybe there should be some specific interaction. And it looks like this TCR is different depending on the company of the vaccination, because HA risk is slightly different between Moderna and Pfizer. So they are similar, but there should be antigen specificity a little bit different, I assume. But again, I don't know the details of your questions.
Okay, another question is genome-driven drug discovery is becoming very popular. And as far as I know, big pharma are very interested in using the biobank genome data for drug discovery. But the genome data is quite sensitive, data transfer. And I'm just wondering whether Japan's government, how to negotiate with big pharma for doing a cooperation for drug discovery.
So Japanese government says okay to share individual genome data within Japan. I think it's also true for other countries. So in this case, we need to negotiate the company, pharma company within Japan, right? So we somehow limit our potentiality for the collaboration. So because we want to collaborate with big farmers globally, right? And this is one of the limitations. But if we want to expand, collaboration with much bigger pharma, but it's something still difficult. But
Thank you for your wonderful talk, Professor Okada. In your slide about the viral integration sites in the telomere of RLE, would you think there is a link between the location of the viral integration and the onset age of the disease?
We don't know because we only, in Japanese population, human HHV is only integrated in 20 to chromosome 22q arm. So actually we don't know other example. But it looks like this, some research says that the integral site is different in Europeans. So I think different. So when we evaluate cross-populational comparison, maybe we can understand, but yeah, it's very small number of individuals who have such integration, so we don't know the details yet.
Thank you so much.
Okay, any questions? Okay.
Okay, so thanks for your inspiring talk. I'm also curious about the clonal hematopoiesis, that part, because you suggest that the somatic mutation in the B cell can affect the maybe the immunity against the SARS-CoV-2. So do you think that is also important, both important, maybe the repertoire status and also the somatic profile in our body with the aging?
Yeah, I fully agree. So, but yeah, again, because there's only limited number of cases who have somatic mutation, and we try to expand our catalog, right? So expand hundreds of individuals who have somatic mutation and single cell. Well, maybe you can get some landscape, but we have very limited combination results. So I agree. I have the same question, but yeah, we still on the way to explore.
And thanks for your insightful presentation. I have some pure question on this topic, because the immune status will affect the vaccine titers or efficacy. So if we can modulate the patients or subjects' immune status before they accept the vaccine to transform the patient to be the potential responder?
Of course, we found that some small number, very small people, but they are significantly impaired vaccine titers. So yeah, maybe we can be able to stratify, or we can pinpoint the people who are less likely to be fully vaccinated. Okay, you are likely to be fully vaccinated, less likely, so you should be more efforts to vaccination. But vaccination things is still sometimes socially sensitive, so it's not fully okay now to stratify the people based on stage. But in pure genetics, we can stratify the population into antibody vaccination responses.
Okay, if there's no any questions, we have to close the section here. Thank you very much. Thank you. Thank you.
Thank you, Professor Okada. If you would be so kind to stay on stage, because we would like the moderator to present you with gifts and a certificate to commemorate your visits, yes. And also to have a commemorative photo of today's occasion. Round of applause. Now it's time for a short tea break. Refreshments and snacks are available outside. Please return to your seats by 10:30 for the next talk. Take this time to relax and socialize. Thank you. Welcome back, everyone. I hope you enjoyed the snacks and refreshments. Let's continue on with our next session. To moderate our next presentation, let us invite Professor Wei Jian Huang, Associate Dean of the College of Pharmacy, to introduce our next speaker. Let's give him a round of applause.
Okay, good morning, everyone. It's my great honor to be here to introduce Professor Tiago Asai.
Yes.
Okay, yeah. Professor Asai is working as a full professor at the Tohoku University in Japan. And he is a natural product chemist, yeah. But unlike the traditional natural product chemist, he used synthetic, biosynthetic approach and genome mining for investigation of natural product. Yeah. And Professor Asai, he has published many outstanding publications in journal. Yeah. Yeah, as we know, such as the English Me and the natural communication and the organic letters. Yeah. And today he will present his ongoing effort by developing privileged natural product scaffold by using synthetic biology. Yeah. Ladies and gentlemen, yeah, please join me in welcoming Professor Asai. Thank you.
Thank you very much for very kind introduction. And I'm Teigo Asai from Tohoku University. First of all, I'd like to thank all the organizers to give me a great opportunity to show our research. Okay, this is my background. So my background is chemistry, natural product chemistry. Especially discovery. So I started from plant natural products, focused on plant surface microstructures, as shown here. And then I changed the material from plant to fungi and focused on activating fungal site biosynthesis cluster. And recently we focus on fungal gene resources by using genome mining. You know, natural products produced by a variety of microorganisms such as plants and microorganisms, fungi, actinomycete, are very important and attractive source for drug development. There is only example. So there are too many excellent natural products that contribute to drug development. So to show all examples. So among natural product producers, we focus on fungi as main resource for natural product study, as shown here. And fungi also well-known producer of a variety of natural products in terms of chemical structures and biological activities. So this is an example of pharmaceutically important fungal natural product. And fungi produce toxin as well, and variety of biological active natural products as shown here. So fungal natural products are really important and attractive source for drug development. So it is still important to discover natural products with new structures and biological activities. Okay, I'll show you an overview of natural product biosynthesis by using penicillin as example of natural product biosynthesis of fungi. Okay, you know, penicillium fungi produces penicillin, but in vivo system. So penicillin biosynthesized from very common amino acids, 3 amino acids, valine, cysteine, and amino adipic acid. So the 3 amino acids are connected in the specific order to give the linear tripeptide by a non-ribosomal peptide synthase, NRPS. And then dioxygenase forms the critical structures for the antibacterial activity in penicillin. So beta-lactam ring and thiazolidine ring as well. Finally, acetyltransferase exchange the N-terminal moiety from aminoadipic acid to phenylacetyl group to give penicillin. So this is biosynthetic pathway of penicillin. And this pathway is based on the biosynthetic enzymes. And biosynthetic enzymes are encoded by biosynthetic gene. And generally, fungal natural product biosynthetic gene responsible for the same natural product tend to form clusters. This is biosynthetic gene clusters. So natural product biosynthetic pathway is programmed in biosynthetic gene clusters. So natural product biosynthesis begin with the expression of biosynthetic gene cluster to form the enzymes, biosynthetic enzymes, followed by building biosynthetic pathway. As a result, natural product structures are formed through biosynthetic pathway. So, in other words, the information of natural product structures is programmed in biosynthetic gene clusters. So, understanding variety of natural product biosynthetic machinery and accumulating those information, we can conduct genome mining to connect natural product structures and biosynthetic gene cluster. So by using genome mining, we can find biosynthetic gene clusters according to individual purpose from the both structural information and genetic information. So I showed that genome mining enables us to access to biosynthetic gene clusters from the both structural information and genetic information. But that said, biosynthetic gene cluster is just information. So we should materialize those information to substances, so natural products. So heterologous expression system is very powerful tool to enable this. So we can get natural product programmed in biosynthetic gene cluster by using heterologous expression. Fortunately, excellent heterologous expression system has been established in fungi. So heterologous biosynthesis of lots of natural products, even if with very highly complex structures that difficult to chemically synthesize, have been achieved so far. So synthetic biology approach based on genome mining and heterologous expression has become a game changer of natural product research. So you know, genomic information is just text. So we translate those information to biosynthetic information by using genome mining tool, such as anti-smash and second find, and so on. And recently, excellent and user-friendly tools have been released one after another. So we always use these tools. And this is typical composition of natural product biosynthetic gene clusters. So most natural products biosynthesis begin with skeletal formation. So biosynthetic gene responsible for the skeletal formation always exists in a natural product biosynthetic gene cluster, except for the case of natural product biosynthesis utilizing ready-to-use primary metabolite as its skeleton. So these are representative skeletal forming enzyme in fungal biosynthesis: polyketide synthase, PKS, non-ribosomal peptide synthase, NRPS, and PKS NRPS hybrid, and terpene cyclase, and so on. So these skeletal forming enzyme genes become a good landmark for genome mining of natural product biosynthetic gene clusters. So I show you an example. So this is a genomic region containing a biosynthetic gene cluster and put in the data into the second find and analyze the region. So the result returned like this. So you can see here PKS exists and some other oxidase around here. So we can recognize this cluster to be natural product biosynthetic gene cluster. So okay, this is an example of genome mining and heterologous expression of acetylcholine. Acetylcholine is one of the antivirus natural products. So in terms of natural product biosynthesis, acetylcholine is a member of diterpene. So diterpene, so the cluster corresponding to the acetylcholine need to include GGPP synthase and terpene cyclase at least, and some other oxidase as well. So based on the biosynthetic hypothesis, so we look for terpene cyclase in the acetylcholine producer gene and find several terpene cyclase on the genome, and then analyze the region containing each terpene cyclase around here, here. So this is one of the region of including terpene cyclase. So in the same manner, by using second find and analyze, and the result returns. And you can see this cluster containing terpene cyclase, and near here, GGPP synthase exists in this region. And this cluster possesses 2 P450 oxidase. So this composition is consistent with the speculation from the biosynthesis aspect. So this cluster is a potential candidate for acetylcholine biosynthetic gene clusters. So once we can, picking up a candidate biosynthetic gene clusters, we conduct functional analysis of the cluster by using heterologous expression. So according to biosynthetic manner, we introduce biosynthetic gene step by step to reconstruct the biosynthetic gene cluster and the pathway as shown here. So introduced first 2 enzymes into aspergillus oryza. Aspergillus oryza is a common heterologous host for fungal natural product biosynthesis. And this transformant produces these compounds. So at this stage, so we realize this cluster to be a biosynthetic gene cluster for acetylcholine because same scale, these share the same skeleton. And continue to introduction of biosynthetic gene step by step and finish reconstruction of biosynthetic pathway in heterologous host. So by using this approach, we can get natural product programmed in the biosynthetic pathway and its intermediate as well. And we can identify the function of all the enzyme in this cluster. So it's very powerful too. This method can be also applied to unknown biosynthetic gene cluster. So find the biosynthetic gene cluster, but at this point, we can not predict the structure of product, only the genomic information. And after we recognize it is possible to produce new natural products, or this cluster show new biosynthesis, we try to conduct heterologous expression. So first, imagine a biosynthetic pathway from cluster composition, and according to the hypothesis, imagine we heterogeneously expressed biosynthetic genes, as shown here, like this, and to get natural product. So if we apply this strategy to unknown cluster, we can find new natural products and identify its biosynthetic pathway as well. So we have changed the strategy to discover natural product from traditional way. So from bioresources to natural product to genome mining as shown here. So first we analyze the genomic information and analyze the genomic information and find the Bayesian gene cluster for our purpose and combine the use of genome mining and heterologous expression to find the natural product and biosynthesis as shown here. So to increase our resources, so we have collected more than 100 species of fungi from plants and insects as shown here, and we have analyzed those draft genomic data when we have extra budget. And increasing the diversity of fungal resources, we gathered a variety of fungi from many collaborators, and we also have analyzed their draft genomic data in the same manner of our collections. So accumulating the draft genomic data and construct our in-house genomic database, now it contains more than 150 fungal species. So to enhance our originality, we usually use our own draft genomic data for our genome mining as a starting point. And we expand it to open database. These genetic information, we aim to find new natural products, variable natural products and their analogs, and the new biosynthetic enzyme and new biosynthetic machinery by using heterologous expression. So today I'd like to talk about the two topics, genome mining of fungal HRPKS and reconstruction and redesign of fungal dietary penoid pathway. Okay, so highly reducing polypeptide synthase is specifically conserved in fungi. And HRPKS is a member of PKS and classified into iterative type 1 PKS. And HRPKS generate a diversity of fungal natural product carbon frameworks. And there are huge number of HRPKS in fungal genome. So even one strain has more than 20 HRPKS. So many HRPKS are existing in fungi. And most of them have been untapped, so unknown. The function is unknown. So we focus on highly reducing polypeptide synthase for our genome mining project. The 3D structure of HRPKS is highly similar to that of mammalian fatty acid synthase as shown here. So the carbon chain constructing mechanism of HRPKS resembles that of M-FAS as well. However, M-FAS solely produces saturated C16 or C18 fatty acid as shown here. But on the other hand, each HRPKS produces different polypeptide chain in carbon numbers and modifications. And HRPKS generally lacks C-terminal domain responsible for the chain releasing from its acyl carrier protein. So M-FAS has thioesterase domain at C-terminal position, and this domain responsible for the chain releasing from fast ACP to generate free fatty acid. But HRPKS doesn't have those the C-terminal releasing domain. So HRPKS usually use partner enzyme as their releasing system. And the type of releasing system related to the kind of natural product classes. This is an example. So for example, so this HRPKS doesn't have partner enzyme. This type of HRPKS produce pyrrole type compounds. And this HRPKS worked with this type of NRPKS with C-terminal thioesterase domain to form resveratrol type macrolacton ring type natural product. So HRPKS, so the diversity of generated by HRPKS depends on the diversity of carbon chain structures programmed in HRPKS and the diversity of partner enzyme for chain releasing. So, so HRPKS, there are many HRPKS. So we try to classify the HRPKS. So we collected the amino acid sequence of more than 10,000 HRPKS from open database and our own database, and carried sequence similarity network analysis, as shown here, after analyzing with different thresholds. So for example, threshold 30%. So it's too condensed, so we cannot classify it. So change the threshold to 50%, pretty good. And threshold change to 70%, it's too fragmented. So we apply the threshold of 50%, as shown here. And based on the analysis of non-HRPKS, we realized that HRPKS tend to make craze depends on the kind of partner enzyme for chain releasing system rather than carbon chain structures programming HRPKS. So in other words, these crates reflect the compound classes. So we focused on untapped crates, explore new natural product and new biosynthetic machinery as well. So first, we focus on this crate. So this crate of color is gone, but so this crate clusters with thioesterase. And this is thioesterase clusters with HRPKS, and these thioesterases are different from known ones in phylogenetic analysis. So, and the cluster composition were rich in diversity, so we hoped to find a variety of natural products and identify new biosynthetic machinery leading to new genomic and leading to new genome mining tool. So this is the first example. Very simple cluster composed from only HRPKS and T thioesterase. Find in the genome isolated from hairy caterpillar. So we heterogeneously expressed both genes in Aspergillus oryzae. The transformant started to produce compounds. And after isolation and structuralization, we determined the structure of the compound 1 to be shown here. So this macrolide compounds possess a 34-member ring and polyene moiety and polyol moiety. It's very unique. So this compound has 7 chiral centers. So we determined that all the configuration of the 7 chiral centers were by using various chemical approach, as shown here. It's very tough work, but we successfully determined all the stereochemistry, as shown here, and cleared the biosynthetic machinery of this type of macrolide biosynthesis. So from these results, so we the results suggested that HRPKS with thioesterase, this crate responsible for the biosynthesis of macrolide. So we continue to functional investigation of this cluster and find a variety of new macrolides with different carbon numbers and modification patterns, as shown here. So, but there are so many clusters in this crate encoding macrolide natural products. So we would like to know what kind of macrolides exist in this crate and how diversity of ring size, but we cannot predict from the amino acid sequence information. So we try to predict the ring size from the sequence, amino acid sequence information. So macrolide natural product. So first, HRPKS produced its own polypeptide chain, and this polypeptide chain transferred to thioesterase, and thioesterase catalyzes macrocyclization, as shown here. And the ring size, so this macrolide has 12 and 34, 22. The ring size macrolide depends on the corresponding HRPKS and thioesterase, as shown here. So in our study, in our biosynthesis study of referral gene. So we successfully get the crystal structure of thioesters. So from the crystal structures, the active site specifically recognizes the polypeptide chain in cyclization form. The structure of the pocket, so in other words, the structure of pocket is correlated with the producing ring structures. So it means thioester serve as a template of a macrolide structure. So we try to develop the prediction model by using the structural information of thioesters. And so by two approaches using machine learning and so structural information of the enzyme. So I just give the idea of this type of research, but this super smart student successfully developed the system. So first, we collected amino acid sequence of the thioesters, and we grouped together thioesters that show highly similarity to each other, so over 80%, and converted amino acid information into structural information through ESM2. EMS2, a kind of protein 3D structure prediction model like AlphaFold. And after that, so we performed machine learning based on functional known TEs, like this. And we successfully classified thioesters as according to ring size as shown here. But so we predict the ring size of thioester ring to known ones as like this, so as color spots. So it is possible. But thioesters, so the ring size so of these gray spots, we cannot predict the ring size of these thioesters isolated from known ones in this model. So we try to develop a de novo prediction system by using more specific active pocket structure information. So because this model developed by my students, so I cannot say detail, but in summary, we extracted the active pocket information from each alpha model of thioesters and compared the size each other and calculated the correspondence to the difference in the ring size. So we try to verify the accuracy of the model by wet experiment. So by using these thioesters highlighted in red. So NRS cluster, so this one. So amino acid homology, we can recognize no signal correlation between each other. But this model predicted these thioesters may produce 40-membered ring. And the heterologous, so we conducted heterologous expression of the thioesters and the corresponding HRPKs in phosphorylase. The transformant produced this compound and isolation and structuralization to show this compound has 40-membered ring. So pretty good. And then these two, so Ka41 cluster. So this is also showing no significant correlation between amino acid sequence, but this model predicted these thioesters may produce 24-membered ring. So heterologous expression of the thioesters and the corresponding HRPKS revealed this cluster produce 24-membered ring macrolide, as shown here. So it's good. So next one, IWLJ cluster. This cluster resembles to APMLB. So very big size macrolide. And so this prediction model predicted despite one unit difference, but the model guessed answer almost right. So heterologous expression shows heterologous expression, and the transformant produce 32-membered ring macrolide. So very near. So we successfully developed the prediction model. So, and by using the model suggested the existence of macrolide with unprecedented ring size. So for example, 18 and 12. So these ring sizes have never been reported from the nature. So we try to obtain macrolide with unprecedented ring size by heterologous expression them. So now ongoing project. So we try to do functional analysis of other clades, such as HRPKS with type 3 PKS. We successfully found new natural product and new biosynthesis as shown here. And HRPKS with C-domain like enzyme also successfully found new natural depth peptide and new biosynthesis. And recently we found very quite unique KS domain catalyzed beta-lactam formation in high-migration biosynthesis. This study demonstrated so typical domain of HRPKS catalyzed atypical beta-lactam formation. So this is summary of genome mining of HRPKS. So through the genome mining of HRPKS, we successfully found a variety of new natural product and a new biosynthetic pathway, and new biosynthetic machinery. And recently we used the macrolide produced by synthetic biology method as synthetic units to produce peptide and polypeptide hybrid natural product like compound library as shown here. So our target, not only HRPKS, but also something so this type of non-canonical PKS to find very unique new chalcone synthase of fungi and NRPS system and PKS NRPS system, PKS NRPS hybrid system as shown here. Okay, this is genome mining topic. So next, we move to reconstruction and redesign of a fungal melatonin pathway. Fungal diterpenoid pyrrole is one of the melatonoids of the fungal natural product and show a variety of biological activity, even though they show similar structures as shown here. The biosynthesis of this type of natural product partially suggested as shown here. And first, the 5 genes highlighted in red color catalyzes the biosynthesis of common intermediate as shown here. Based on the information, we mined the fungal diterpenoid pyrrole biosynthetic gene cluster, and we found 5 types of biosynthetic gene cluster as shown here: FG, MP, CH, ASMA clusters. So we reordered based on amino acid sequence similarity and function as shown here. So all the clusters share the first 5 genes as shown here. So we heterogonously express all the enzymes in the heterologous aspargillosylases and demonstrate all the clusters share the common intermediate. So like this. And so we analyze the modification steps as shown here and predict of the biosynthesis pathway. So for example, the 3 clusters FG, MP, and CH share the two SDR type oxidase as shown here. So this means they may share the pathway until here. On the other hand, ASMA has an individual modification enzyme, so they may be separated at this point to give final products. And CH pathway possesses different enzymes from these two clusters, so this pathway branched at this point to give final product. And these two pathways branched this point to give final product. So based on the hypothetical biosynthetic pathway, we reconstructed biosynthetic pathway in asparagine as shown here. So first we introduced 5 enzymes to produce this common intermediate, and step by step we introduced enzyme to produce this intermediate of the pathway. And then we introduced the final enzyme into the appropriate host to produce the final product of each pathway as shown here. So through the reconstruction of the biosynthetic pathway, we found new intermediates and new diterpenoid pyrrole natural product as shown here. So analysis of the function of the enzyme in this pathway. So the enzyme shown in orange box catalyzes C5 friendly unit as shown here. On the other hand, enzymes shown in green box catalyzes on pyrrole moiety as shown here. But no pathways include both modification enzymes. So we try to artificial pathway by redesign of the biosynthetic pathway. So we added this enzyme to this pathway, and we added this enzyme to this pathway to construct artificial pathway. Habanat program in nature. So this is the result. So we heterogeneously expressed in the additional enzyme into this host to produce non-natural natural products as shown here. So of course, this combination of the cluster has not been reported from nature. So all the compounds had new structures as shown here. So we, through the reconstruction and redesign of biosynthetic pathway, by using these 5 gene clusters as materials to form 5 natural pathways and 5 artificial pathways as shown here, to form 22 diterpenoid pyrrones, including 12 new diterpenoid pyrrones produced in this study. So this study demonstrated this strategy is very powerful tool to expand the chemical diversity around the biological active natural product. So we then evaluated their biological activities, and we found anti-ATRB active diterpenoid pyrrones, and some diterpenoid pyrrones showed innate immune suppression activities, and some diterpenoid pyrrones show inhibition of amyloid beta aggregation, and growth inhibition of cancer stem cell, like this. So we successfully produced a variety of diterpenoid pyrrole compounds by using reconstruction and redesign of fungal melanoid pathway based on synthetic biology method, and found a variety of biological active natural products. Okay, this is the last slide. So now our laboratory tried to final goal is find of new drug seeds. So we concentrate to try genome mining and biosynthesis study, and sometimes so reconstruction of compound library by using biosynthesized natural product. So finally, so I'd like to thank all the collaborators and all the lab members. So thank you for kind attention.
Okay, thank you so much, Professor Asai, for his ongoing effort for developing the novel natural products by using structural biology. It's a very wonderful work. Yeah. Are there any questions to Professor Asai? Okay. Or Professor Xie, please.
Thank you, Professor Asai. It's brilliant work that you developed this systematic tools to predict the size stories that generate different ring size of macrolide. I'm just curious, do you observe any intermediate enzyme that lacks the specificity for the ring size, so therefore they generate a mixture of macrolide in different ring sizes? Do you see that? Because you are able to generate 34 ring, which is amazing. But I'm just wondering, did you observe a single product, or you see a mixture?
So some enzyme produced 1 or 2 ring size.
Yep.
But almost, so the combination of the HRPKS TE produced specific ring size and specific modification patterns.
Mm-hmm.
Yeah.
Yeah, but do you see any enzyme that has no specificity and is able to generate a ring size of, say, let's say, 12, 14, 16?
You mean this?
Yeah, the...
Yeah. So these HRPK is different from each other.
Yeah.
So active site structures is different from each other. So the active site structures maybe reflect the structures.
Okay.
Yeah, but so we didn't know how regulate HRPKS produced its own chain length. Yeah, we can understand pseudoestrus served as a template, but we can understand how regulation of the elongation pattern of the HRPKS. It's very difficult point.
Yeah, that also brings the second question regarding to, you mentioned that you have a software that is similar to AlphaFold, but it's called EM.
Yes.
Can you tell me a bit more about that? Because I don't, yeah, and I personally never heard that before, so I'm just wondering if you can introduce to us what exactly this.
This one?
Yeah.
Very similar, but so accuracy is low than AlphaFold, but the speed is higher. So first we use ESM2, but now we use AlphaFold.
Okay.
Okay. Yeah. So because of machine spec.
Yeah.
Yes,
Okay. Cool. Great. Thank you very much indeed. Thank you. Wonderful work.
Thank you, Professor Asai, for an interesting talk. My question is very practical. So today, do we use Aspergillus species organism as expression host now for express this fungal enzyme, or still we can use Saccharomyces, the model organism, to do the same biosynthesis pathway discovery?
Yes. So our laboratory usually use Aspergillus oryzae as host because Aspergillus oryzae is national fungi in Japan, and so whole genome already analyzed, and so I have one more reason because the researcher in Tohoku University developed the Aspergillus oryzae heterologous expression system, so we learned from him the handling of the Aspergillus oryzae, so we use Aspergillus oryzae. But so some US group and Chinese group use Aspergillus nigrans as a host, and some groups use Saccharomyces as host for fungal natural product. But I think so for fungal natural product, it's better to use fungi, same fungi. So Aspergillus oryzae or nigrans is better, I think. And Aspergillus oryzae has potential to express the many enzymes. So Aspergillus oryzae have high potential to produce natural products and natural product biosynthetic enzyme. So it's very good, excellent host. So we use.
Okay. My second question is about, for example, when you find the cluster and you have, for example, for the oxido reductase, you have two P450 homologs. So how do you know their function? So you still need to do an empirically experiment to check their function to see which enzyme do what oxidation on which.
Now we cannot predict from the sequence. So we. So as I said, we can recognize, so just this enzyme joint P450, but we cannot predict the function. So what kind of substrate this enzyme recognizes and which position this enzyme oxidizes, we cannot predict. So we should wet experiment, so heterologous expression or in vitro enzymatic analysis, enzymatic reactions.
So one last question, possible? Yes. So you say that we can discover new natural product using when we discover the biosynthesis cluster. That means before maybe some natural product we have not yet identified. So using your genome mining, to provide a tool to discover new natural product.
Yes.
Yes.
So so to discover new natural product, we mine the cluster which doesn't show similarity to non-biosynthetic cluster, or so we imagine the cluster, what kind of compounds and what kind of oxidation stage, and we search the reported natural product and compare the cluster and the structure and find and recognize the possibility to produce new natural product or not. But we cannot predict perfectly. So some case, so after heterologous expression, the biosynthesis gene cluster, we think this cluster to produce new natural product. But after that, we find non-natural product. But even if the case, we find new natural biosynthetic pathway. So both are okay.
Thank you.
Okay. Since time limited, I will conclude this. Okay, let's go. Okay, if there are no question, I will conclude this talk. Yeah, and thank Professor Asai for his talk. Yeah, once again.
Thank you very much.
Thank you, Professor Asai, for answering those questions. Again, if we could have you stay on stage, because we will be giving you gifts for your visit here. And if the moderator could present the gifts to the presenter. Thank you very much. Now we invite Professor Yo Wenzhen, Associate Dean of the College of Pharmacy, to moderate our next presentation and introduce the speaker. Let's give her a round of applause.
Thank you. Good morning, everyone. It's my honor to introduce our next speaker and my dear colleague at the School of Pharmacy, Taipei Medical University, Dr. Changjun Wu. Dr. Wu is both a physician and a bioinformatics scientist. He earned his MD at the National Taiwan University and his PhD in bioinformatics at Boston University. Before joining TMU, he spent many years in the United States, including at the University of Texas Anderson Cancer Center, leading major cancer genomic and immunotherapy projects. He also worked in industry as a senior machine learning software engineer at Meta, a very famous company, building large-scale deep learning systems at Layer. And also, his publication was published in many journals such as Nature, Nature Cancer, and Nature Communication. This publication highlights his contribution to precision oncology and AI-driven biomedical research. Today, he will share his latest work on how AI-driven T-cell receptor signature can help predict which lung cancer patient will respond to immunotherapy. So please join me in welcome my colleague, Dr. Changjun Wu. Yeah, thank you.
Thank you so much for the kind introduction. And yeah, and I hope everyone enjoyed the talk today. And so before my talk, I would like to thanks for this invitation. And yeah, so after like more than 20 years in the United States, I'm so fortunate to be back to Taiwan and can join the TMU pharmacy as a full-time faculty. And so, because of my experience, in the past decade, I have been deeply involved in and also have hands-on experience in many of the buzzwords, like from the bioinformatics, like big data analysis, precision medicine, or cancer immunotherapy, and AI. So today, so today I'm going to introduce one of my recent publications. Applying AI in the T-cell analysis in lung cancer. So we know that the T-cell is the central of the cellular immunity against like pathogen or like a tumor's mutation. Yeah. So the most important part is the T cells, like extracellular receptor or TCR. So TCR is a heterodimer. So most commonly is alpha chain and the beta chain. So and each chain has 3 complementarity determining regions. And the most important is CDR3. And because CDR3 directly bind to the antigen, so it may be more important in the specificity. And the antigen is usually presented by the MHC molecules, and then bind to the T-cell receptor. And you know that to tackle the almost infinite number of the antigens, so the T-cell had to be hugely diversified. And how does this T-cells sequences come from? So you know that we have the VJ segment on the alpha chain or the VDJ segments on the beta chain. So during the maturation and the development of the T-cells, these segments can recombine, rearrange, and splice, and as well as in the addition of random nucleotide to make this T-cells very, very diversified. And we call this entirety of these T-cells as the T-cell receptor as the TCR repertoire. So the size of the TCR repertoire is about 10 to the power of 12 per individual, or 10 to the power of 19. And TCR sequencing is a great tool, powerful tools to profile the TCR sequences. So usually you can do the single cell TCR sequence. In this way, you can profile the alpha chain and the beta chain together. But the throughput is not high, usually maybe thousands to tens of thousands of the clones is available. Or you can do the bulk sequencing. Then the throughput may be up to maybe 100,000. But usually you can only do alpha chain or beta chain, not both. And usually the beta chain is more popular because, partly because the beta chain may provide more diversity to this repertoire. Yeah. And the project I'm going to introduce, actually, we have two aims. So one is, if we can see, if we can distinguish the lung cancer relevant or specific T cells or TCR sequences from the data. And the next aim is that can we develop TCR biomarkers or signatures that allows to predict the immune checkpoint blockade treatment response in non-small cell lung cancer. And this project has been published very, very early this year in the European Journal of Immunology. Yeah. If we are able to achieve these two aims, then we may have better extend to other topics. Maybe better be able to design like CAR T cell therapy, or to develop like cancer vaccines or vaccine to other disease, or we may use this signature to find the right people and the right immunotherapy for cancer treatment. However, there are some challenges of that. So the first challenge is that the TCR repertoire is very, very large. And the majority of this repertoire, actually, they are like a background TCR T cells. And this T cell repertoire, actually, they are dynamically changing from day by day, or the aging, or with the onset or progression of the diseases. And although the lung cancer antigen, the disease-related antigen, is maybe only a small fraction, but the proportion of these specific antigens, T cells, actually we don't have no idea how many of them. And the other major challenge is that if we study the TCR sequences among like different individuals or people, we found that the overlapping rate at the sequence level, no matter it's the full sequence or the CDR3 sequence, is awfully low. So the overlapping rate can be as low as like 0.1% or below. Yeah. Then this disables us from grouping or clustering those T cell receptors for further analysis. Then how to resolve this kind of challenge? Okay, the technical challenge first. Then of course, from my title, we have to use AI, the artificial intelligence models. So Dr. Asai just introduced the language models. So this part of the AI models, they adapted from the old like natural language processing models. Usually, this part we use the transformers. So we can map the sequences to continuous numeric vectors. This kind of modeling, it can capture semantic meaning in the relationship between the sentences. By the semantic meaning, we mean like what this word is talking about, what is the topic of this word. So we call it the contextualized word embedding. The idea is that in this high-dimensional embedding space, maybe 64 or 100 and more, the words with similar context or similar meaning, they'll be closer in the space, in the distance. So the resolution is low, but these are the menses, these are some verbs and the noun about the talking. Yeah. With this word embedding or embedding at the word level, they can be aggregated to have an embedding of the sentence. Then the sentence embedding can be further aggregated to the text embedding, such that the model can tell us what an article is saying. Yeah. So applied to the biological research, people developed some protein language or embedding models. and also on the TCR embedding. There are a couple of TCR embedding models. Okay, can convert the TCR sequences to the embedding space. So the idea is that, so the first step of our project, actually we apply this kind of the TCR models to convert all the TCR sequences into a numeric space. So first we collect a very, very large collection of the non-disease cohort as our background. So using this kind of the TCR2-back model, we can embed each beta chain, V plus CDR3 plus J segments to a 120 dimension vector. Then, because we are more used to work on a two-dimensional space or the landscape, so we further apply the principal component analysis to project all these embedding vectors onto, we call it a TCR embedding landscape, using the top two principal components. So in this way, each sentence, now they become one point on the landscape. And to further cluster or group these TCR sequences, so we consider, so we further partition the whole landscape into roughly 2.5 million grids, basically on the x-axis and the y-axis, each had 1,600 equally sized bins. Yeah. The idea is that the sequences or the points belong within each grid are considered as one cluster or one group. And we define the density of the grids as the number of the points or the normalized number of the sequences within each grid or cluster. And this yields the background TCR embedding landscape. So this is how this landscape looks like. Yeah. So the darker the blue color, it means that there are higher density or more points or more sequences within each grid. Then when one new cancer cohort or one cancer sample, the TCR data is coming, then we can apply the same approach up to this level. We use the TCR to-back model to convert the sequence into a very big matrix containing the TCR embedding numbers. But we cannot, at this step, we cannot do a principal component analysis from scratch, because in this way, all the coordinates will be changing and they cannot be comparable. But we use the eigenvectors from the previous step to project the converted embedding matrix. Now they are all on the same universal embedding coordinate system. So this is how it looks like. The red ones are the new data, and the gray ones are the background. And you can see that all the red ones, like they sit on the background distributions. Actually, this makes sense because we know that the immunity, the T cells, when they need some T cells to bind to some antigen, they did not invent or create new T cell clones. Actually, they expand existing T cells from circulation and find the one that can react or be activated by this antigen. So it makes sense that actually all these cancer-related grids or groups are from existing ones. There's no really cancer-only or cancer-specific text. But the good thing is that with this embedding approach, now the overlapping rate can be improved from like 0.1%, now it's to between like 11 to 15%, which allows to do the further analysis, which is cross people or cross cohort analysis. Okay, now back to the first challenge. Remember that how can we tell which TCR sequences are cancer specific or cancer relevant, and which TCR sequence might be background or non-cancer related. Or so we have some ideas. So the strategies that first, we collect two lung cancer cohorts which had paired lung primary and brain metastasis TCR data. The idea is that if an antigen or T cell group, a clone, that can be shared in both primary lung, primary side, and the metastatic side, then it is more likely to be disease-related or cancer-related. The opposite may not be true, but I think if the same T cells or the T cell have the same binding targets we're seeing in both the primary and metastatic loci, yeah, it may be disease relevant. So this is our first idea. And the second idea is that we were focusing on the brain metastatic expanded T cells. Why is that? Because we believe that from our clinical and biological insight, we speculate that the primary lung tumor might be more heterogeneous in the T cell distribution because it has more heterogeneous mutations. And also the brain side, the microenvironment in the brain is usually much cleaner and with fewer background T cells. So we use a criteria like we request that the density in the brain of the ratio between brain to lung had to be at least 4-fold. And lastly, the common tricks that to build a good performing signature or biomarker, we hope that the same pattern or the same clone can be found in at least 3 subjects, different subjects. So with this criteria, we end up with 358 tags. We call it anti-LAA tags, anti-lung cancer associated antigen. And these 358 anti-LAA tags include roughly 1,800 TCR sequences in the brain. And yeah, so as a prognosis signature or biomarker, actually it performed pretty well. So this aggregated anti-LAA tags level is significantly higher in the ICB responders than the ICB non-responders. We use an antiviral text as a negative control, and it did show that there is no difference between the responders and the non-responders for this non-specific signature. Yeah. And in addition, this anti-LAA tag levels is significantly positively correlated with the tumor mutational burdens. Yeah, it's like 77%. Very significant p-values. Okay. Then the next question is that what is our next plan? How can we boost our immunogenomic study to the next level? Yeah. So we know that to looking only at the TCR sequence is very limited and not enough. To trigger a good immune response, it needs the cooperation or binding of all like TCR sequences, the peptide and the MHC molecules. So a good strategy should take into account all these 3 components together, not just one. This is our previous work in the COVID-19 study. So in which we studied the binding between the COVID-19 spike protein with the common human MHC molecules. And we look at how the variants in the spike protein can impact the binding affinity or the immunogenicity from the Wuhan variants to Delta, from Wuhan strain to Delta, to Omicron, to XBB1. Yeah. So we have a series of papers published. However, with the new model, like Dr. Asai just mentioned, so with the new structure prediction models like AlphaFold3 or the ESM3, we should be able to study or to get the embedding of this, either the MHC peptide, the antigen peptide, or the TCR peptide at the structure level, we should be able to retrieve the embedding. Yeah, actually, we have a pioneer project on the dengue fever virus just started, and this is a preliminary proposal. So we would like to profile or like a TCR sequences, the HLA genotyping, and the antigen peptide from the dengue virus or other disease antigens. And we would like to propose to use this AI-enabled model to get their embedding at the sequence and the structure level and at the repertoire scale. And then the difficult part that we have to design a new, maybe very complicated, we call the dual tower or the tri-tower deep neural network AI models to predict the binding affinity or the immunogenicity. Yeah. And with all this prediction, then we may be able to do the one we are most interested in, the rational vaccine design. Yeah. Yeah, so a little bit fast. So this is my question, and thanks for your attention. Any questions?
So thanks, Dr. Wu, and thank you. So wonderful presentation. Yeah, it's really a deep learning for me. So difficult. So now we open question for our audience. Okay, please.
Hi, thank you very much. That was a great, great talk and very exciting method. What I was wondering is, do you have, so the signature that you found, could you validate it in new patients that you recruited?
So thanks for this question. It's a very, very important point. Yeah, I should be able to find out, but actually for the training, we have used up all the data, available data we can find online. So. Yeah, so probably you should cooperate with the clinical clinicians and to see if you can.
Yeah, I think that would be very interesting because I think that maybe the difficulty with these approaches is that it's hard to know if it's a bias in your training data set. But yeah, I think if you could demonstrate the same clones, and my next follow-up question was going to be that it might also be nice if you could clone some of these and screen them potentially against peptides to show that it would be a nice full circle.
Yeah, exactly, yes. And one good thing is that for training or developing this model, we need paired lung and brain meta data. But to validate them, actually, we may only need the lung primary data with TCR. It's more easily available.
Yeah.
So is there a question? Oh,
hi.
Thank you.
Thank you for a wonderful presentation and your research findings. Since you mentioned about COVID-19 virus,
yes.
Investigation of the toxicity in that chain, what do you think about the current COVID-19 toxicity? Because in the US, we're thinking that COVID-19 current strains are not that virulent or not toxic anymore. And then that's probably one of the reasons that the President Trump is not going to support the vaccination, including COVID-19. The government is not going to, insurance company is not going to pay for it. We have to pay if we want to get vaccines. So what do you think about the toxicity of the current strains of COVID-19?
Yeah. Thank you very much for this question. Yeah, so we with our dean, Professor Zhang, we study the binding affinity to the variant. So we did observe that maybe the toxicity or severity of this COVID-19 infection kind of decreased with the new strains. But I think this is the meaning of our study, that because we don't know when or how long maybe a new strain will be, will emerge, and then the immune storm or the immune response will be pushed to another level. So I think to know the binding affinity according to the variations or the mutations or evolutions on the spike protein or other protein is very important. Yeah. Any other comment?
So, is there any question? So I think everyone must be very hungry, waiting for the lunch. Okay, thank you, Professor Dr. Wu, to final our morning section, and thank you so much. Yeah, so we end this section. Thank you.
Thank you, Professor Wu, for answering questions. We have prepared gifts for you as well, so if the moderator could present those gifts on stage. Thank you. Okay, thank you very much. Ladies and gentlemen, we will now take a break for lunch. Lunch boxes are provided outside, and so please head to the designated areas as shown on this screen. So for speakers, we have prepared the 15th floor conference room for you to have your lunch. For the audience members, I believe it is the medical building B1 as these rooms shown on the screen. So enjoy your meal, and we'll see you after lunch. Thank you. Ladies and gentlemen, we'll begin our conference very shortly. We'll probably start around 10 past 1, so 1:10 p.m. So if you want, take this time to go to the restroom or get some coffee or drinks, or just sit and relax for a bit. So we'll begin our conference shortly. Thank you. Welcome back, everyone. I hope you had a very enjoyable lunch. We'll now resume the afternoon sessions of the conference. For our next presentation, we have invited Professor Yves Xie, head of the Department of Pharmaceutical Sciences, as our moderator. Please welcome Professor Xie to introduce our speaker today. Thank you.
Thank you very much. Welcome back. So it is my great pleasure to introduce our first speaker this afternoon. Gustav received his Bachelor of Science degree with first-class honors at the University College London, the UCL. And then he spent a year in industry before beginning his doctoral studies at the University of Oxford in late 2023. Under the supervision of Professor Benjamin Fairfax in Department of Oncology. So today, Gustav will share his recent work published in Nature Medicine as a first author, very impressive, entitled From Genetic to Chronic Viral Infection, Novel Insight into Systematic Determinants of Response to Checkpoint Immunotherapy. So please join me in giving a very warm welcome to Gustav.
Thank you very much for that very kind introduction, and also thank you very much for the invitation to come and speak to you all today. I must confess I am not a pharmacist, so I have learnt a great deal over the past 2 days with lots of very insightful talks. So I'm going to try and keep the ball rolling, as we say. And I'm going to talk about a class of therapeutics called immune checkpoint inhibitors. And these have completely transformed the way in which we treat lots of different kinds of cancer. And it was actually the subject of a Nobel Prize. Yes. So as it was said, today I'm going to be discussing systemic determinants of response to this kind of treatment. So I thought I would start by giving a brief history lesson in how we treat melanoma. So we primarily focus on melanoma, and melanoma is often viewed as the greatest success story of immune checkpoint inhibitors. So in this timeline, I would like you to focus mainly on the pre-ICB era. So this is before checkpoint inhibitors were developed. If you had metastatic melanoma, so melanoma that had spread, you were normally treated with alkylating agents such as dacarbazine or high-dose cytokine treatments such as IL-2. And a lot of these treatments were really quite toxic, and actually the benefit was relatively small. So the median overall survival was about 7 months. So if you had metastatic melanoma, unfortunately, this was a completely fatal disease. And then in 2007, the first immune checkpoint blocker was developed. So this was called ipilimumab, and it targeted a checkpoint protein called CTLA4. And then later on, we got the development of anti-PD-1 and anti-LAG3, and various combinations of these treatments. And now we're at a stage where metastatic melanoma patients have a median overall survival beyond 5 years. So going from 7 months to over 5 years, we would argue that this is probably the greatest success in cancer treatment in the last 50 years. So, like I said, immune checkpoint blockers are an incredibly successful treatment, but how does it actually work? And this was quite nicely introduced in the previous talk. But essentially, the premise is that there are these effector, so cytotoxic T cells, that are basically carrying a T cell receptor that is specific to some kind of antigen that is expressed on a tumor cell. And so basically this means that your immune system is able to recognize a cancer cell as a kind of foreign invading agent, and is able to use that TCR to mount a cytotoxic response which then kills that cancer cell. However, in the cases of chronic diseases, and this is true of viruses as well, not just cancers, basically your immune system has evolved a kind of negative feedback mechanism where, to prevent it from overreacting to things and resulting in autoimmunity, what will happen is these inhibitory proteins get upregulated to the cell surface, and they compete for stimulation of this T cell. So PD-1 and CTLA-4 are examples of this. And so essentially what happens is once these blocking receptors, these inhibitory receptors get expressed, the activity of the T cell begins to slow down. And so even though you had the right T cells to fight off that cancer, ultimately you're no longer able to mount a response. And so there's this genius idea of, well, what if we design monoclonal antibodies that block these receptors? And so essentially we give a lifeline to these T cells to carry on signaling and carry on killing cancer cells. And so the trial that a lot of our research is based off is this Checkmate 67 trial, where essentially they assessed patients who were treated with different kinds of checkpoint inhibitors, so ipilimumab, which is the anti-CTLA-4 antibody, or nivolumab, which is an anti-PD-1, and then actually one arm where they were treated with both. And essentially they then followed these patients up for now over 10 years, and they look at their overall survival. And so what was obvious from this trial was that actually the grey line, so those patients treated with the CTLA-4 inhibitors, had the worst survival, and actually blocking PD-1 seemed to be much more effective. But actually you can see that in the combination arm, there is a further benefit. So if I add both drugs, I get a better response. And so what we can see here is that actually 52% of patients were still alive at 5 years following administration of this treatment. So in oncological terms, if you are cancer-free within 5 years, we would say that you're essentially cured. And so, unfortunately, though, like with most immunotherapies, this does come with some risks. So actually, a lot of these patients develop so-called immune-related adverse events. So these are kind of autoimmune-like reactions that you get from the drug. And although the mechanisms driving this aren't really known, the kind of leading hypothesis is that essentially there are T cells that are non-specifically getting activated by the drug, which can then go on and target healthy tissue. So they can develop things like this nasty rash that this patient developed. But actually what's quite interesting with these immune-related adverse events is that several meta-analyses and actually data from our own cohort seem to suggest that patients who get these autoimmune toxicities actually have improved responses to treatment. And so we can see specifically that actually it's these mild, so grade 1 to 2, we tend to class these immune-related adverse events in terms of the severity and also the organs that they are affecting. And we can see that specifically the lower grade toxicities and also ones that are found in the skin, so dermatitis, these are the ones that seem to have the best overall survival benefit. And so potentially what this suggests is if we can understand the mechanisms of immune-related adverse events better, maybe this will give some insight into why patients respond. But so we're left with a number of questions. So despite the fact that I said that this is one of the greatest success stories in cancer treatment, only 50% of melanoma patients get a benefit. And actually, the ones who don't get a benefit seem to progress quite rapidly. And we don't really know why. And there is a need for identifying biomarkers to say which patients are likely to respond and which ones aren't. And equally, we don't really understand why certain patients develop these immune-related adverse events or these toxicities. And so our idea was essentially, rather than focusing on features of the tumor, which often requires taking tumor biopsies, which is quite an invasive procedure, what if we could identify biomarkers from the blood? So it's much easier to get blood samples, you can get these more readily, but actually possibly you can get a lot of information about what's going on in the tumor by looking at the blood. So several studies previously have actually demonstrated that you can identify tumor infiltrating lymphocytes in circulation. So we want to try and identify some of these biomarkers. And then secondarily to this, what we want to determine is, are there factors beyond the tumor? So you've all probably heard of biomarkers such as tumor mutational burden or neoantigen load. So melanoma is often regarded as a very highly mutated cancer, which is why we think that immunotherapy is so effective for it. But is the anti-tumor immune response being shaped by things that are not related to the tumor, so systemic determinants, as we like to say? And so the point of this is essentially, can we then stratify patients and say, well, based on X, Y, Z markers that we find in you, we believe that this specific immunotherapy is the one that you should get. So in this case, I said that the combination of anti-PD-1 and anti-CTLA-4 seems to really increase the risk of developing these nasty side effects. So if we can find patients that will actually get just as much of a benefit from the single agent anti-PD-1 treatment, maybe we can direct them that way. Or maybe we can say, actually, you're someone who would benefit more from a targeted therapy. So patients who, for example, have BRAF mutant melanomas, maybe we would say you should get a BRAF inhibitor instead of a checkpoint. And finally, potentially these peripheral biomarkers could elucidate novel pathways that we could begin to target. So can we design then new therapies that might sensitize those patients who are not responding to checkpoint blockers? And so to address these questions, we've started a cohort study which is now dating back to 2015, and this is called the Oxford Cancer Immunotherapy Toxicity and Efficacy Study. And essentially what we've done is we've prospectively recruited now over 500 patients, primarily melanoma patients, but now we're recruiting across a variety of cancers. And so all of these patients are getting treated with immune checkpoint blockade. And what we then do is we take blood samples from these patients in a longitudinal fashion. So we will take a sample before they get treatment, and then early on in treatment. So day 22 is after they've had 1 cycle of immunotherapy, and then day 63 is after they've had 3 cycles of immunotherapy. And then what we then do is we follow these patients up for a long time afterwards, and we get an indication of their overall survival, but also the types and severity of the immune-related adverse events, so these toxicities that they get. And using these blood samples, what we can do is we can do proteomics assays, and we also do single-cell and bulk transcriptomics on the immune cells that we find in their blood, along with flow cytometry. And then we can integrate all of this kind of deep immune phenotyping data back onto how the patient responds, and we can try and find correlates of outcomes and toxicities. So the first aim was to identify some peripheral biomarkers of response to immunotherapy. So the very first kind of major observation in the lab was actually that we could see that patients who had a larger number of expanded CD8 T cells, so these are T cells that carry the same TCR, but they have expanded in the blood in response to some kind of antigenic stimulus. And what we could see is that the ones in the blue line that had above a median number of these large clones had both improved progression-free survival and overall survival. And then when we assessed these clones in more detail in terms of their transcriptome, so the kinds of genes that they expressed, we could see that the large clones really segregated from the smaller clones, so they were phenotypically quite distinct. And this actually associated with an induction of lots of these cytotoxic genes. So these are the genes that I said are involved in allowing those T cells to kill target cells. And so this made us very interested because we could identify features that were not necessarily tied to the tumor that seemed to predict how a patient was going to respond. And these were quite early indications. So just after 1 cycle of treatment, we had a good sense of how well the patient was going to do. But we wanted to know whether these clones were potentially relevant in the context of melanoma. So we basically took clones, so these T cells that we could find in the blood, and we looked at melanoma tumor samples. And what we could see is that, sorry, this is slightly blurred, but the largest T cells, so those that occupied greater than 2.5% of the circulating repertoire, so of all the T cells that we could find in the blood, they were really, really enriched in melanomas. So it suggested that there was preferential trafficking of these very large clones into the tumor. And actually what we could see is that these effector T cells that we could find in the melanomas, they actually had a greater expression of we called it a cytotoxicity score, but this is essentially just a metric for greater expression of cytotoxic genes. So there's basically large, very active clones that we can find from the periphery in melanoma tumors. And so we then began to wonder whether, actually, is this cytotoxic gene set that we've identified in these clones, is that in and of itself upregulated in patients who have an improved response? And so in bulk RNA sequencing data, we actually found that patients who had an ongoing response at 6 months had elevated cytotoxicity. So their T cells had greater capacity to kill than in patients who progressed. And this was also true at day 21, so just after 1 cycle of treatment. And then the natural question is, well, okay, are you essentially measuring the same thing in 2 different ways? Are you measuring large clones and cytotoxicity, but actually it's the same thing measured 2 different ways? And so what we did is we stratified patients according to the number of large clones that they had, and also the degree of cytotoxicity of their T cells. And interestingly, what we could see was that there's this compensatory effect, where actually the only group that had really distinct survival were the patients who had both a lower amount of cytotoxicity and fewer large clones than you'd want. So that's this group 4 down here. And so essentially this suggested that cytotoxicity was an independent peripheral biomarker for response. And so we've now identified 2 features of T cells that you can find in the blood that seem to associate with response. But this isn't necessarily demonstrating some kind of causality. So, you know, what are these clones doing? Are they just a correlate? You know, what are the features that are resulting in some patients having more of these kind of cytotoxic T cells? And so what we began to wonder was, well, are there drivers beyond the tumor? So not something dictated by the kinds of metastases they have or the tumor mutational burden, but something potentially beyond that. And so we first decided to look at germline genetics, and it's been illustrated very nicely this morning, but the impact of genetics on your immune responses is incredibly well known. So I think the best example is looking at the MHC or the HLA region of the genome, which is the most polymorphic. The idea being that essentially across a population we're protected from a wide array of pathogens because we each have a slightly distinct capacity to present certain peptides to our immune system. And so what we then began to wonder is, well, we know that from lots of studies that almost all autoimmune diseases have very strong genetic associations. And like I said, these patients tend to develop these autoimmune-like reactions to treatment. So we began to wonder, well, are there genetic variants that associate with the likelihood of developing these immune-related adverse events on treatment? And then can we use that to understand something more about the biology behind how the immune checkpoint blockers are working? And so to do this, we performed the first genome-wide association study in collaboration with the Dana-Farber Cancer Institute in Boston. And we basically took over 1,700 patients who were treated with single-agent PD-1 or PD-L1, and we basically looked at their risks of developing, after follow-up, of developing immune-related adverse events. And what we could see was that there were 3 risk loci that were associated with increased risk of developing a toxicity. And so one was in IL-22 receptor, one was in a long non-coding RNA, and one was intronic to this gene IL-7, which we were particularly interested in because IL-7 is a crucial cytokine in lymphocyte survival and also in T-cell maturation. So we decided to focus on this polymorphism here. And so what we first did was we looked at the genotyping data that we have from Oxide, so in our cohort, and we actually saw that the effect replicated. So particularly in those treated with anti-PD-1 alone, if you had the alternative allele at this locus, we could actually see that there was a much greater likelihood of patients requiring steroids. So these are immunosuppressants for these toxicities that they get. But what we wanted to leverage our cohort for specifically was to try and understand how was this polymorphism acting? Why was this genetic variant associated with increased risk of immune-related adverse events? And so we started from a very basic perspective of, well, what is the role of IL-7 in melanoma patients? Is it a crucial cytokine in some capacity in our patients? And so what we did was we basically took, we did bulk RNA sequencing of IL-7 across a range of immune cells from blood, and we did this for healthy donors and patients, and then we compared the expression. And what was immediately obvious was that actually IL-7 was really quite profoundly elevated in B cells, so these CD19 lymphocytes. And specifically, we could see that actually the patients had a further induction of B cell IL-7. So it suggested that the IL-7 that was being produced by B cells was somehow important in the immune response to melanoma. And so we then wondered, okay, well, we know that B cell IL-7 specifically seems to be important, so potentially this polymorphism is acting on B cell IL-7. So when we repeated this analysis, but we stratified for carriage of the risk alleles, so these are the patients who are more likely to get the autoimmune side effects, we could see that actually they had a further induction of IL-7 relative to other patients. And although this was trending in the same direction in the healthy donors, we didn't see a significant effect. So essentially what this highlighted was that this polymorphism was an EQTL, or an expression quantitative trait locus. So essentially these are polymorphisms that either regulate the expression of genes in the local area, so cis-acting EQTLs, and there are others which actually are trans-acting, so a carriage of a polymorphism can regulate gene expression in a distal part of the genome. And so what was then the immediate next question was, okay, so we know that this polymorphism has this specific effect on B cell IL-7, and so actually, is B cell IL-7 really the driver behind the increased risk of toxicity? So what we did was we repeated our analysis for predicting the risk of immune-related adverse events, and we actually found that B cell IL-7 across all genotypes was predictive of increased risk of toxicity. So the more B cell IL-7 you had, the more likely you were to develop a toxicity. And when we corrected for the expression of this gene, we could actually see that the effect of the polymorphism disappeared. So what I think is really elegant in this example and reveals some of the power of genetics is essentially you can take a genetic variant that regulates the expression of a gene, and we know that specifically it is the effect that it's having on this gene that is driving some kind of phenotype. So we can kind of follow this causal chain. And so like I said, we were originally quite interested in IL-7 because it has reported effects, obviously, in T-cell survival and maturation. So we wanted to see whether this B-cell IL-7 or carriage of this polymorphism was doing anything to the T-cell compartment as well. And so what we could see was that actually there was a trend towards an induction in the proportion of these effector, so TEMRA, terminally differentiated effector T cells, and a really pronounced reduction in the proportion of naive T cells in the patients who carried the risk allele. So it suggested that indeed carriage of this polymorphism was resulting in increased T cell maturation. And likewise, revisiting the idea of clonal expansion, we could see that across treatment types, patients who had the risk allele seemed to have more of their T-cell repertoire occupied by these large T-cell clones, so those occupying greater than 0.5% of the circulating repertoire. And so finally, we demonstrated that this polymorphism interacts with treatment. So if you have the polymorphism, you have slightly different responses to treatment in terms of toxicity. But we then wanted to take a step back and say, well, we know that these patients have elevated IL-7 before they even get treatment. And does that potentially have some kind of impact on the development of the melanomas that they get? So using the TCGA data, we looked at patients who had earlier stage melanoma, so stage 2 or stage 3 resectable melanomas, and this was over about 400 patients. And interestingly, what we could see is that carriers of the risk allele actually had improved disease-specific survival, so they were less likely to go on to get metastatic disease. And so essentially, in summary,
this polymorphism identifies a group of patients who are at increased risk of getting a toxicity on treatment in the metastatic setting, but it actually seems to regulate the kind of the development of the melanoma itself. And so the next question was, well, your genetics obviously has a profound impact on your immune responses from birth. But a lot of the time, when patients have developed cancer, they often do this, they get this later in life. And so often these patients have had decades of exposure to various kinds of pathogens, which each in some way have left their mark on the immune system. And while some pathogens, for example, influenza, will result in a kind of sharp immune response, which then calms down over time, some patients are infected with these chronic viral infections, which have a much larger footprint on the immune response. So examples of this are Epstein-Barr virus or cytomegalovirus. And so we began to wonder that, well, given that we know that some of our cohort will have these chronic infections in the background, maybe this is also having an influence on their clinical outcomes. And so to do this, we focused on cytomegalovirus, or CMV specifically, and we did this for a number of reasons, but the first reason was that actually in the UK, which is where our study is based, about 50% of people are CMV seropositive. I believe in Taiwan it's probably more like 90% or 95%, but in the UK we were quite well powered to detect any kind of difference CMV might be making. And also, viruses like EBV have long been implicated in cancer development, so things such as Burkitt's lymphoma have been tied to EBV. But actually, CMV has been rather omitted from the field of medical oncology, and it's mostly focused on in the context of congenital CMV in neonates, or in patients who've undergone an organ transplantation or who have HIV and are immunocompromised. So we thought that it might be interesting to explore CMV from this point of view as well. And so CMV is a rather odd virus because actually when you get infected with it, you're infected for life. But rather than having a kind of consistent activation, it's a latent chronic viral infection. So essentially what happens is you'll get I think the lasers died. But you get an initial spike of kind of viral replication, and then over time you will get these small reactivation events where you get small amounts of viral replication. And we were specifically interested in this phenomenon of latent infection because of the impact that it has on T cells. So what we see that actually seems to be quite specific to CMV is you get this effect called memory T cell inflation. So if you look at the blue line here, essentially what happens is T cells that are specific to CMV, so they have a CMV reactive TCR, they tend to actually continue to expand over time. So the same T cell clone can be identified oftentimes several years later, and the amount of space that it occupies in the T cell repertoire will kind of slowly shift upwards. And this is in stark contrast to acute infections where if you were infected with the flu, I would expect that in 3 days' time you will have lots of flu-reactive T cells, but then potentially in 2 or 3 months they would occupy quite a small amount of the T cell repertoire. And finally, several studies, but quite famously this one by Simone et al., actually demonstrated that a lot of the tumor-infiltrating lymphocytes that we find in patients seem to be cross-reactive to CMV. So they seem to have some kind of specificity against CMV. But actually their role as passive bystanders or as active agents in the anti-tumor immune response is not particularly well described. So the first thing we decided to do was to see, can we replicate the effects of memory inflation in patients who have cancer? So a lot of these studies that I described previously were done in healthy individuals or in mice. And so what we did was we isolated CD8 T cells from patients before they got treated, and we stratified them according to CMV positive and CMV negative patients. And we could see that there were several hundred differentially expressed genes across these patient groups. And quite specifically, we could see that there was induction of genes associated with tissue residency. So ZNF683 and CCL5, these are crucial in essentially increasing the trafficking of T cells from the blood to the tissues. But we could also see that there was elevated expression of cytotoxic genes. So these are the ones that I said are involved in CD8 T cell mediated killing. And when we then did a pathway analysis to try and get a better understanding of what these genes were contributing to, we could see that there were several pathways that were upregulated in CMV positive patients. These are the ones in orange. And specifically, we could see there was increased peptide presentation on MHC. There was increased interferon gamma signaling and also increased TCR signaling. And so all of these pathways are really, really crucial in both the antiviral response but also the anti-cancer response. And so to then see what kind of T cells these genes were coming from, we performed blinded flow cytometry to CMV status, and we could find that there was a really, really profound expansion of these TEMRA, which are the highly differentiated T cells in the CMV positive patients. So this gene set was contributing, was resulting from an expansion of these cells. And so all of this is quite well known, but the flow data revealed something else which was of interest to us, which was actually that these patients seem to have a contraction in the proportion of the activated, so CD25 is a marker of activation of these activated Tregs. And so Tregs are known... Oh, thank you so much. Great, thank you so much. So what we could see is that there was a really significant reduction in the number of these regulatory T cells in CMV positive patients. And the reason why this was interesting is that Tregs are known to be negatively prognostic in a lot of cancers, but specifically in melanoma. And we then began to wonder, well, okay, maybe CMV is having some kind of impact on the way that patients respond. And so what we decided to do first was, given that we saw this induction of cytotoxic markers in our CMV positive patients, we used the cytotoxicity score, which I described previously was a predictor of improved outcome. And we basically tracked how this evolved in patients in different treatment arms, but also according to their CMV status. And what we can see consistently is that CMV positive patients have elevated cytotoxicity relative to the CMV negative patients. But interestingly, in the CMV negative group, there seems to be a divergence in the response depending on the kind of treatment that was given. So in the combination treated patients, we can actually see that there's quite a significant induction of the cytotoxicity score after they've been given treatment, but you don't see this in the single agent treated group. And so we then began to wonder whether this had potential clinical implications. So when we looked in our cohort of patients who were treated with the combination anti-PD-1 and anti-CTLA-4, we could actually see that CMV had no effect on overall survival at all. So these two lines are almost perfectly superimposed. But when we looked at those patients who were treated with anti-PD-1 alone, we could actually see that there was a significant overall survival benefit in being CMV positive. So this was really interesting to us because it was the first indication that potentially an environmental factor, a chronic viral infection, was altering the way in which you respond to cancer treatment. And so actually, we, to follow best practice, we assessed this in a multivariable analysis. So we corrected for things like age, which is quite strongly associated with CMV status, and also sex and the kind of melanoma, so BRAF mutant or wild type. And furthermore, we replicated this finding in a smaller cohort of patients. Sorry, the numbers are very small here, but this is a smaller cohort of patients who are treated in the adjuvant setting. So this is for stage 2 or stage 3 resectable melanomas. All these were in the metastatic setting. And we could actually see that, again, CMV positive patients who received anti-PD-1 had improved recurrence-free survival. So it suggested that CMV was interacting with anti-PD-1 treatment specifically, but there was some kind of compensation that was going on in the combination treatment. And so seeing as we could demonstrate that essentially cytotoxicity score and CMV by virtue of this was predictive of different outcomes, we were wondering whether this gene set that CMV was influencing in CD8 T cells, whether this had any core transcriptional drivers. And so basically in the using bioinformatic techniques, we could identify 9 transcription factors that had increased activity in our CMV positive cohort. And then we basically assessed each one to see how well it correlated with the CMV differentially expressed genes that I showed in an earlier slide. And what we could see was that actually this transcription factor TBX21, which encodes TBET, which is the master regulator of all kinds of immune responses when you think of interferon gamma or granzyme B, these type 1 immune responses. This is the master regulator of that. And we could see that this one was the most strongly associated with our CMV phenotype, which we collapsed into this CMV score. And to further add to this, in our single cell data, we could identify effector T cells that carried a CMV reactive TCR, and we could see that TBX21 was much more highly expressed in the CMV-specific T cells relative to those which were non-CMV specific. And we also demonstrated this in mice. So with mice that were infected with a mouse strain of CMV, we could see that T cells that were reactive to this epitope called M38 had much higher chromatin accessibility at TBX21 compared to naive T cells or central memory T cells. So this again suggested that TBX21 was crucial in this CD8 T cell response to CMV. But what were the implications on clinical outcomes? So what we were hoping is that, well, potentially if we can identify a causal transcription factor for this gene set that is influencing the way in which CMV interacts with treatment, we might find potentially a novel pathway that we can intervene in some therapeutic capacity. And so, interestingly, what we then found was that actually, if you just looked at TBX21 expression in CD8 T cells on treatment, those patients who had higher TBX21 had a significantly improved overall survival. So if you had low TBX21, you had a hazard of death of 2. So really, these patients were doing much worse. And so potentially this indicates that TBET is a mechanism that we can potentially intervene in and design some kind of new therapeutics. And so, like I said at the start of the talk, we know that your response to immune checkpoint blockade in terms of your overall survival and also the toxicities that you develop on treatment are quite tightly associated. So we then wanted to see how CMV was changing the autoimmune-like reactions that these patients were getting. And rather to our surprise, we actually found that if you were CMV seropositive, you had a much lower likelihood of developing these grade 3 and above. So these are the most severe toxicities that patients get. So these toxicities often require systemic steroids. They often require patients to come off their immunotherapy. So these are really, really nasty. And we could see that in the orange line, CMV positive patients had a much lower cumulative incidence of these toxicities, which is kind of contrary in some ways to what I was explaining earlier, because I was saying that autoimmunity was a good thing in terms of your overall survival, but actually we find that specifically these high-grade toxicities aren't necessarily beneficial to have. And then we wanted to dig a little bit deeper to see whether this actually associated with different kinds of organs that were being affected. And actually what we could see was that CMV was having pleiotropic effects. So if you were CMV seropositive, you were less likely to develop colitis, pneumonitis, or myalgia, which is kind of general muscle pain, but you were more likely to develop dermatitis, which are these skin toxicities that I said were quite predictive in improved overall survival. So it suggested that, again, an environmental factor is altering the kinds of autoimmune reactions that you get on a cancer treatment. And then finally, we thought, well, similarly to the IL-7 polymorphism that I described in the previous section, we know that CMV modulates your immune response, and specifically that it modulates your immune response in the context of cancer immunotherapy, but was it doing anything to the development of the tumor in the first place? So using the UK Biobank, which was mentioned earlier, we basically performed age and sex matching of patients in the UK Biobank with a CMV status and our cohort. And we could find, interestingly, that across all metastatic melanomas, so all groups, and then specifically in BRAF wild type and BRAF mutant melanomas, CMV seropositive patients were slightly underrepresented to what you would expect by chance. So there were fewer CMV positive patients. And so this suggested that maybe CMV seropositivity was having some kind of protective effect against developing metastatic disease. And although you can't see this effect in the adjuvant melanoma, so the stage 2-3 resectable melanomas, which is a kind of less advanced disease, you can tell that by the confidence intervals here that we had much fewer numbers. So we're trying to kind of assess this now in larger studies. And also, secondarily, we know that the epidemiology of these BRAF wild type and BRAF mutant melanomas are quite different. So you have a greater risk of getting brain metastases in BRAF mutant disease, and they often tend to present slightly younger. And so we decided to look at the age of presentation, or in our case, the age at which the patient turned up for systemic treatment. And we basically divided the cohort according to their BRAF status and also whether they were CMV positive or negative. And interestingly, we found that actually in the BRAF mutant group, there was no age difference at all between CMV negative and CMV positive. But actually in the wild type CMV positive patients were on average 9 years older. So it meant that there was some kind of delay in the presentation of metastatic disease in these CMV positive patients. And so the obvious question you might ask now is, well, okay, this is great, but how is CMV doing this? And this has now become a really, really big focus in our lab. So some of the hypotheses that we have I've presented here. So like I said, CMV reactive T cells have been demonstrated to be found in tumors. And so potentially, actually, these aren't passive bystanders. Maybe they're actively contributing to cancer cell death, or maybe they are secreting cytokines downstream of TBX21 that can basically induce activity in neighboring tumor-reactive T cells. And also, like I said, maybe it's just shifting the balance in your immune response towards a more effector-like phenotype with reduced immunosuppression. And so, in conclusion, I hope that I've convinced you that actually host genetics and infection history have a profound effect on your response to cancer treatment. And actually, we need to expand our view beyond just looking at inside the tumor and features of the tumor in trying to predict how a patient will respond. And actually, in some ways, I don't think this is that surprising because, like I said at the start of the talk, checkpoint blockers work on existing immune responses. So these are patients that have a certain immune makeup to begin with, and that immune makeup has been shaped by a variety of pathogens that these patients have been exposed to, and also kind of what they were dealt at birth. And so in some way, I think it allows us to revisit this idea of the cancer immune duel. So as we know, the tumor and your immune system are locked in this kind of evolutionary arms race. And I like to visualize this as a game of chess. So every time your immune system makes a certain move, the cancer will make some other move to try and evade it. And I think that that's all well and good, but actually all of this is happening in the context of your genetics and environmental exposures. And so you can almost imagine that the dimensions of the chessboard and the different moves that each side can make are really, really biased by these things. And finally, I think, or my hope is that I've shown that kind of large data and careful phenotyping of these patients really facilitates translational research. And I think that to benefit our patients, I really hope that more cohorts such as this will begin to emerge. And finally, I just want to reference some of the studies that I've discussed today. And then I want to thank a long list of people who have contributed to this work and our sponsors over the last 10 years. And specifically, I want to thank the patients and their families, because without them, none of this research would be possible. So thank you very much.
Thank you very much. It's very eye-opening. Questions from audience? Because I do have a lot of questions, but I'm actually giving you guys a chance to ask questions. Yep.
Oh, hello. Thank you for your comprehensive talk on cancer treatment, immune checkpoint blockade, ICB, and the role of chronic CMV infection in T-cell modulation and therapy response. And in some low latitude countries, there are many kinds of viral disease, such as yellow fever and dengue. And although these are typically considered acute viral infections, but some patients may still experience the antibody dependent enhancement, ADE, either upon reinfection or even during the primary infection. And this suggesting that their immune system has been altered. So my question is, could such acute viral infections also influence the tumor development or affect responses to cancer therapies? And I would greatly appreciate your opinion. Thank you.
Thank you. Yeah, that's a great question. I think the difficulty with the short answer is I think so. You also find lots of influenza-specific T cells in tumors, and I've never really bought this argument that T cells are in a certain place but are doing absolutely nothing. I think in terms of the kind of tumor microenvironment, they will definitely be relevant. I think it's much harder to track, though, because with acute infections, it's difficult to know potentially when the patient was infected. There are more kind of confounders that emerge. So the nice thing with chronic infections is that we know that they're still kind of mounting a continuous CMV response. But yeah, I think it would be very interesting to explore. And I don't think many people have looked at that at all.
Any other questions? Yep.
Okay, thank you for your outstanding speak. And I have I'm wondering to know we know the pembrolizumab is a ICI, right? And recently the clinical trial is focusing on this ICI combine another cancer vaccine like mRNA 4157, this Moderna mRNA. I want to know if the mRNA segment contain CMV antigens. It also can boost the ICI efficacy.
Yes. So this is actually this is very interesting, because I think one of the questions I often get asked is, okay, so are you saying that everyone should be infected with CMV? I would say no, don't infect yourself with CMV. But I think that, and there are companies that are now trying to do this, that understanding the memory inflation response that you get in CMV is something that people are trying to use in cancer vaccines, because the idea is that you're essentially getting like a supercharged T cell population, and if you could direct that against some tumor-associated antigen or neoantigen, you could make a great vaccine. So there are people who I think are looking potentially at integrating tumor-associated mRNA in a kind of CMV backbone or something like that. But yeah, it's a great point.
Thank you.
Any other questions? So oh yeah.
Okay, thanks for your wonderful talk. I just have one question about the TCR part. Because you are focusing on the CMV and the TCR, and both we know that CMV contribute the epitope, and the HLA is very important, and TCR can recognize it. So are you planning to identify what kind of TCR are most important and what the target they recognize in the future?
Yes. So
thank you.
so kind of off the back of the, and this is kind of unpublished data, but off the back of the idea that we see that these large clones are specifically predictive of improved response, we can actually also see that patients who have a very good response, they often have a few T-cell clones that years later we can find them in their blood and they're still really, really expanded. And these are often enriched in CMV positive patients. And so the natural kind of theory is, well, they must be CMV specific, but we don't actually know. And so I've started a project where we're trying to identify some of these clones that we think are important, and we're basically screening them on libraries of peptides to try and identify what they might be targeting. Because I think what's really interesting, and this is more on a technical note, but essentially we know that T cells are really cross-reactive, so they can see not just one antigen, but they can often see hundreds or thousands of different antigens. And so while I don't necessarily think that it's the systemic determinant of how CMV is impacting response, if you think about it, if you had one or two cross-reactive events in a single patient, some of these T cells are so large that actually you wouldn't need so many examples of this for it to have a big effect. If suddenly 2% of your repertoire that comes from one clone can be directed against the cancer, that's already a huge asset. So I think it's very interesting to find out if there's any crossover between TCRs being able to recognize CMV epitopes and also cancer epitopes.
So we have time for one more question. Anyone from audience? Yep.
Hey, wait. Hello.
Hey.
Thank you for your wonderful talk. I'm curious, does the protective effect on CMV positive on ICB efficacy depend on specific HLA alleles? For example, HLA A2, B7, which are known to be associated with CMV response. Thank you.
Good question. So in our data, we don't seem to see that there is a HLA effect, specifically not for those alleles. The difficulty is with rarer alleles, your power starts getting much smaller, but with A2 and B7, we don't really see much. But what is interesting is we do see that carriage of certain alleles seems to alter the magnitude of this memory inflation response that you get. Yeah, so I think that possibly, almost certainly HLA will have a part to play. But I think, again, we just need more numbers to say that conclusively.
Now the time's up. Let's give another round of applause to Gustav. Thank you very much.
All right, Mr. Gustav, if you could stay on stage for a little bit as we have our moderator, Professor Xie, to present you with a gift as token for your visit. Thank you. Thank you very much. Next, let us have Jiali Han, Chair of the Master Program in Clinical Genomics and Proteomics, to join us as moderator for this presentation. Let's give her a very warm welcome.
Good afternoon, everybody. Are you still awake? It's my pleasure to introduce the next speaker, Dr. Wanxuan Zhou. Dr. Zhou received her full education training in Taipei Medical University, including bachelor degree in pharmacy, master degree in clinical pharmacogenomics and pharmacoproteomics, and PhD degree in School of Pharmacy under the supervision of Professor Wei Chaozhang. Dr. Zhou is currently serving as a pharmacist in Taipei Medical University Hospital. She is also a certified genetic cancer risk assessment specialist trained in City of Hope. Because of her excellent academic performance, Dr. Zhou has received several scholarships, travel grants, and poster awards in international or domestic conferences. In addition, she actively participates in the pharmacy-related association, including the Pharmacogenomics Society of Taiwan, Taipei Pharmacist Association, Taipei Young Pharmacist Group, and International Pharmaceutical Students Federation. Today, she is going to share with us her point of view in harnessing genomic information in precision oncology. Please join me to welcome Dr. Wanxuan Zhou.
Thank you very much for your kind introduction. Good afternoon, everyone. My name is Wanxuan Zhou, and I'm now a pharmacist, and also I'm providing genetic cancer risk assessment at Taipei Medical University. And as introduced, I'm also serve as a Deputy Secretary General at the Pharmacogenomics Society of Taiwan. So today I'm going to talk about harnessing genomic information for precision cancer prevention, detection, and therapy. So as the advancement of genomic technology, it has now been implemented in across the cancer patient's journey and bringing benefits to all of these steps, starting from the cancer risk stratification, screening of cancer, prognosis and diagnosis, and also the treatment selection and monitoring. So today I will walk you through all, this whole journey, and let's see how, what and how genomic technologies bring benefits to the patients in their different stages. So as we know that cancer is a genomic disease, all our cells can accumulate genetic damages, genetic changes like mutations when we grow old. And if some of the mutations are occurred in very important genes, especially for those regulating cell growth or death, it may drive the tumorigenesis. And for those genetic changes that occurred later in the life and confined only to the tumor cells, we call them somatic mutations. And we can study them through tumor biopsies or liquid biopsies, and we will cover that in our later slides. And these informations are especially informative for cancer detection, treatment, and monitoring. However, before all this tumorigenesis occurs, we also notice that some of our patients are born with genetic mutations that make them more likely to get certain type of cancers. And of course, most of the cancers, around 80% of them are sporadic. The majority of them are sporadic like this. But around 10 to 15% of the cancers are known to be hereditary. It means that the patients can inherit some cancer risk mutations from their parents, making them more prone to getting cancers. And some infamous examples like TP53 and the HR genes like BRCA1 or BRCA2, and the mismatch repair genes like MLH1 or MSH2. And for those patients, these born genetic mutations are presented not only in the tumor cells, but also in the normal cells, just in every cell in the body. And for this kind of mutations, we call them germline mutation. It's just from the germline. And we usually study them, we can study them through a blood draw or barcode swab. And this kind of genetic information is especially informative for cancer risk stratification, and now with a PARP inhibitor, it can sometimes have implications for treatment selection. So what does it mean to have a hereditary cancer syndrome. So as we know that in the model of cancer development, the patient may accumulate the genetic mutations, and then finally it becomes a tumor. So you can think of these patients with hereditary cancer syndromes start their journey of mutation accumulation earlier. They may carry some of them when they were born. So for the sporadic the patients with, for example, let's take an example of tumor suppressor genes as example. For patients with sporadic cancer, they may born with no mutations on their germline, and they may get the first mutation on one copy of their gene, but it does not make the gene function defect. However, when they get the second hit later in their life, the gene can become loss of function and lead the cell to the tumorigenesis. And for the patients with hereditary cancer, they naturally carry the first hit in their germline, in every cell in their bodies. So they only have to acquire an additional somatic mutation on their cell, then it will go into tumorigenesis. So the cancer of the patients with hereditary cancer syndromes are characterized by with early onset, multiprimaries, and they often cluster in families. So what does it mean to have a high-risk genetic mutations? Let's take BRCA1 as an example. For patients carrying BRCA1 or BRCA2 mutations, they have higher risk of developing breast, ovarian, pancreatic, or prostate cancers comparing to the general populations. Like the BRCA1 positive patients, they may have the lifetime risk of getting breast cancer is around 60 to 72%, while in general population it is around 13%. But what does it mean for the patients to know about this kind of mutations? First of all, they can know their risk of developing cancer, and by knowing this kind of risk, they can take some actions to prevent their future cancers, or they can increase their frequencies of health checkup so that they can detect the cancers earlier. And it can also help them to adjust their family plans for reproduction, lifestyle changing, career planning, or even being prepared financially for their probable cancer in the future. So a famous example is Angelina Jolie. She had a strong family history of breast cancer and decided to have both of her breast and both of her ovary removed when she found out that she's a BRCA1 mutation. So as per the current guideline, those BRCA positives, they will be recommended for different kind of breast checkup starting from different ages, and they will need to consider and discuss about some risk reduction surgeries. And for the prevention of ovary cancer risk, if the patient is in their reproductive age, the reproductive plans will need to be taken into consideration in the whole discussion. And she will also need to regularly check her pancreatic and melanoma risks. Okay. So in addition to those single gene that is high penetrance for cancer risk, like BRCA, scientists are also identifying some risk variants that are that have a lower penetrance and have a smaller effect on the cancer risk. So let's think of the risk of developing cancer, like how dark the room is. So if you, with a BRCA1 mutation, it is like to have a large bulb burn out in the room. So the room will be really dark. The risk of getting cancer will be increased for BRCA mutation patients. However, for the rest of the genes, having one of them burned out does not make a lot of difference in how dark the room is, on how risky the patients would get cancer. But having many of them out, having many of these kind of mutations, it will make a big difference in a higher risk of cancer. So now scientists are also aggregating this effect of cancer risk and try to calculate a score, calling polygenic risk score, to estimate the overall genetic risk for the patient's cancer risk. Now we also have some computer algorithm available out there where you can input the patient's personal details, their lifestyle, whether or not they are smokers or drinkers, or you can also input their reproductive histories and their previous breast screening results, and also their genetic results like the PRS, and very importantly, their family history and whether or not they have been tested for those high penetrance genetic mutations. And after input this kind of data, you can estimate the patient's lifetime risk of getting breast cancer, and it can also help you to predict the probability of a patient that may carry a certain high penetrance genetic mutations. This kind of information is especially useful when you try to discuss with your patients about their risk of getting cancer, getting certain kind of cancer in 1 year, in 5 years, or in the rest of their lives. And they can also compare their own cancer risk with the general populations, because different people have different values on the risk. Some people may think the increase of 5% is really a large increased risk, but some other people just don't think about it. So it can help you to discuss with the patients about their own risk and add into their own value on this about the results. Okay, so now we already know that germline mutations are very informative for cancer risk stratification. You can help your patients to find out their own cancer risk, and they can take some actions to try to prevent future cancers or prepared in advance for the possible cancer in the future. So next, move forward to cancer detection. We can also use the genomic technologies to detect possible cancer in the bodies. And from this step, we are more concerned about the somatic mutations that can be found in the cancer in the tumor cells. Okay, so normally when we try to detect cancer, we rely on the image test, but sometimes the tumor is just not big enough to be visualized in the image test. So now with the genomic technology, we can study something called circulating tumor DNA. The idea is that if there is a tumor inside the body, the tumor cells will grow and die just like normal cells. And when a tumor cell dies, it will release some DNA into the circulation. And those circulating tumor DNA can be studied from a single blood draw, and we can look into the serum and see and study the epigenetic or genetic patterns of this ctDNA. So here's an example of Pathfinder study, which is a prospective cohort study just published recently. in this study, the authors, they screen the patients using a multi-cancer early detection blood test, and for the participants over 50 years old. And the MCED blood test, it can detect the cancer signal by studying the methylation pattern in the ctDNA. And furthermore, it can also help predict where the cancer is probably origin from by looking into the patterns. So across in this cohort, across 6,600 participants screened, 92 of them were tested positive in the MCED test. And after a 1-year follow-up, around 38 of them actually were confirmed with cancers. And for most of them, they were detected negative in the MCED test. And for those detected negative, they actually, over 98% of them did not develop cancer in the following 1 year. So for those detected positive and actually have confirmed cancers, 85% of the time that the blood test can accurately predict the original cancer site. So let's take a closer look at those tested positive on the right. Some of them are actually true positive. They have confirmed cancer in 1 year, but some of them are false positives. So as you can see here, the true positive basically took less than 2 months to get their cancers confirmed, while the false positives had to spend nearly half a year to make sure that actually they did not have the cancer. And what's really relieving here is that most of the false positives only need the lab or image test to get the reassurance of not getting cancer. But around 1/3 of them had to have invasive procedure like biopsy or even surgery to make sure that they actually did not have cancers. So the question here is that the harm and the benefit balance of this screening are still need to be studied in the future. And another thing for this test here is that the overall sensitivity of this test is around 29% amongst the 121 cancers that is confirmed in the 1-year follow-up time. Only 35 of them were detected by the MCED. And this kind of sensitivity is just a little bit better than the standard screening strategy in the US. So further studies are needed to find out if there is really an added value of this MCED test to the existing screening methods. And another thing here, when we look at this, we can also think of the cancer type, cancer stage of cancer that they really pick up. So most of the cancer picked up by the MCED blood test are in their later stages. Only 6 out of the 29 cancers were in their earlier stages. So future studies are also needed to find out the benefit of this MCED test on the overall population mortality. And for a new cancer screening tools like this, some other factors need to be considered, like the testing frequencies required, and also the cost effectiveness of this kind of test. Although there is still many questions to be answered, I'm positive that we will see more of this coming in the future. And I believe that with the advancement of the technology, this kind of test will become more accurate. Okay, so we now know that the genomic technology is useful for cancer detection, but when there is really a cancer and a confirmed cancer case, we need to try to treat the cancers. And it is also a very applicable field for the genomic technologies. So when it comes to the cancer treatment, both the somatic cancer genome and the germline host genomes are very important because the somatic cancer genome, the profiling of cancer genome can help us tell how the cancer is different from the normal cells, so we can find the treatable target, and this profile is associated with the treatment's efficacy. And on the other hand, the host genome, especially the pharmacogenetics in our body, it tells us how the drug is metabolized in the body, and it is also associated with the toxicity of the treatment. So let's start with somatic cancer genome profiling. It is typically done in tumor biopsy, but as I said, the liquid biopsy, in a liquid biopsy, we also have somatic cancer genome information available. And these kind of cancer genome profiling may come into a variety of sizes. It can be a single gene panel that include only the one genetic mutation that you are interested in, that has like a BRAF mutation or something. Or it can include, it can be a hotspot and multi-gene panel, which includes multiple genes that you can look into predict the response of multiple treatment agents at the same time. Or it can be a more comprehensive panel that include a lot more genes in a large panel, and can be a whole exome or whole genome sequencing. And now our national health insurance in Taiwan has covered the next generation sequencing panel for some of the cancers. And actually the reimbursement is based on the number of genes included in the NGS panels. The smaller panels, as defined by Taiwan NHI, the smaller panels is those include less than 100 genes, while the larger one is those has at least 100 genes. So they are reimbursed with different price. Okay, so when it comes to the cancer genome profiling, I said that both tumor biopsy and liquid biopsy can be used to study the cancer genome. However, the tumor biopsy is still the golden standard for most of our current clinical scenarios because of its it is irreplaceable place for tumor histology and staging. In addition, in a tumor biopsy, we can study a variety of biomarkers, including the DNA, RNA, and proteins, while the ctDNA analysis are restricted only to the DNA. And another thing is that the liquid biopsy is very vulnerable to the tumor shedding rate. Some of the tissues just don't like to release DNA into the circulation, so it may be more prone to false negative results. That being said, the liquid biopsy that we can take just from a blood draw, it is undoubtedly less invasive than the tumor biopsy. And sometimes the liquid biopsy is the only option available, especially when the tumor biopsy is not obtainable. For example, there are some situations like if the tumor is already removed from the surgery, it is very hard to monitor the disease through tumor biopsy. So we can study the minimal residual disease through the ctDNA analysis. On the other hand, we can do a serial measurement. We can take the blood draw for many times and to monitor if there is a treatment-resistant cancer developing. Okay, so when we do a cancer genome profiling, we are typically looking into the genomic biomarkers like gene mutations, fusions, or a larger extent of the change like microsatellite instability or tumor mutational burden. And some of the biomarkers are specific to certain cancer type, but some of them are called tumor agnostic biomarkers, meaning that they are predictive for treatment responses regardless of their cancer original site. So here are some examples for tumor agnostic genomic biomarkers, like the BRAF mutation, RET, NTRK, FGFR fusions for the target therapies, or the MSI or TMB for the immune checkpoint blocker. Now the ASCO and ESMO both recommends to do the genomic testing for advanced solid tumors, especially for the countries or the place, the region that have tumor agnostic therapies available. Like in Taiwan, we have these kind of therapies available. So, so we already know that the cancer genome profiling is very useful in cancer treatment selection. However, let's take rewind a little bit. When the tumor are still small, they are in their earlier stage, we can usually remove them through surgery. But as I said, when the tumor is removed by surgery, it is really hard to monitor the disease through tumor biopsy. So it is when the ctDNA analysis come into play. So here is the study design of dynamic trial. In this trial, the scientists recruit patients with stage 2 colon cancer and randomize them into the ctDNA guided group or the standard of care group. And in the ctDNA guided group, the patients were tested for their post-operative ctDNA, and the decision on the adjuvant chemotherapy after the operation depends on the ctDNA results. For those tested positive, they will receive adjuvant chemotherapy. Well, for those tested negative, the scientists will hold the chemotherapy and for observation. So overall, in this study, you can see that the proportion of patients receiving adjuvant chemotherapy, the proportion has reduced by 50% in the ctDNA guided group compared to the standard treatment group. So here is they recently released their 5 years follow-up results. So they found that although there is a reduction of chemo use in the ctDNA guided group, it actually does not compromise the treatment efficacy in terms of overall survival or recurrence-free survival. And they also found out that actually the survivorship is associated with the post-operative ctDNA. For those tested negative in the post-operative ctDNA, they actually have a very low recurrence rate after 5 years. So I don't know how you think about these results, but as a pharmacist, when I see this kind of result, I really feel exciting about it, because if we have a way to precisely find out which patient, some of the patients actually do not need the treatment, and if we can precisely identify them through some kind of test like the ctDNA, and we can hold the treatment, and we can naturally prevent them from getting the treatment-related toxicities. So I was really excited about this results when I saw this. And on the other hand, in addition to the somatic cancer genomics, the germline genomics, especially the pharmacogenomics, also played an important role in the cancer treatment. Pharmacogenomics basically describe how our body, how the drugs interact with the gene and proteins inside our body. After the administration of the drug, it can be absorbed by our body and distributed to the treatment target site and sometimes to the unwanted target sites. And then it can be metabolized into different molecular forms and then excreted by the body. So across all these procedures, the drugs will interact with a variety of proteins inside our bodies, like the transporters, the receptors, or the metabolizing enzymes. So if there is a genetic change that alters the function of these proteins involved, it may change the blood concentration of the drug inside our body, or it can change the body's sensitivity to the drug. And here are some examples of the anti-cancer drugs that with known pharmacogenomic interactions. For example, the thiopurines like mercaptopurine, it can be metabolized by TPMT and not T15. And for the patients that have lower activity, that have genetic mutations, so that they have a lower activity on either of the enzymes, they can have a higher blood concentration of active metabolites, and leading to a higher risk of cytotoxicity that may have higher risk of developing severe neutropenia. So now we have already have a recommendation, dosing recommendation available in CPIC on the 6MP based on the TPMT and not T15 phenotypes. For example, the TPMT poor metabolizers, meaning that they have a very poor activity on the enzyme, they will have to have a largely reduced starting dose of 6MP. And now the clinical practice guideline also recommend testing for TPMT and not T15, especially in East Asians, because that we have higher frequencies of not T15 intermediate metabolizers. The next example is irinotecan, which can be metabolized by UGT1A1 into inactive form. So for those with lower activity of UGT1A1, they may have accumulated concentration of the active metabolites and have a higher risk of irinotecan-induced diarrhea or neutropenia. And now there's a dosing recommendation saying that the UGT1A1 poor metabolizers could have a reduced starting dose could reduce their starting dose by 30% for their treatment. However, although there is a dosing recommendation available out there, the current clinical practice guidelines still haven't come to the consensus when we should do the UGT1A1 testing. So I think with accumulation of future evidence and consensus in a different expert group, we will know better which is the best timing for the UGT1A1 testing. And then here comes the fluoropyrimidine and the DPYD. The capecitabine and 5-FU can be transformed into inactive form by DPYD, and those with lower activity of DPYD, like those of poor metabolizers, they will have higher concentrations of active metabolites, and leading to a higher risk of treatment toxicity. And we also have dosing recommendation available out there to reduce the dose based on the DPYD phenotype. However, currently the US FDA and NCCN withhold the recommendation of the universal pretreatment DPYD genotyping. It is mainly because that there are still a lot of unknown about the DPYD variants. Some of the variants we still don't know how it can affect the function of the DPYD. And another thing is that it is still uncertain that the dose reduction really does not compromise the clinical efficacy. So I think that it may be more logical for the DPYD testing and PGX-based dose reduction for those taking capecitabine for palliative purposes, but not for the curative settings. But because in the curative settings, maybe we may be more tolerable to the toxicity, we just want to make sure the clinical efficacy. So in addition to the anti-cancer in addition to the anti-cancer drugs, the pharmacogenetics has implications on a variety of supportive care in the context of oncology, like for the painkillers, the antiemetics, the antidepressants, and also the anesthetics that are usually used in the surgery. So with all these genomic technologies available in the clinical settings, we in TMU hospitals, including the Wanfang and Shuanghe hospitals, we have now have several precision medicinal services available in our own hospitals. For example, we have a counseling clinic for genetic cancer risk assessment. In this clinic, I will take the patient's family history and carefully, and then review their family history and their chart, and refer them for germline testing whenever appropriate. And when the genetic test come back, I will discuss with the patients about their own cancer risk and what they or their family can do with this risk. And sometimes we may recommend their family members to do additional germline testing to know their own cancer risks. And furthermore, we also have a molecular tumor board in our hospitals. In these molecular tumor boards, we regularly have meetings of regularly have multidisciplinary meetings. We have genetic counselors, oncologists, pathologists, radiologists, pharmacists, and bioinformaticians who are majorly in charge of analyzing the genetic data. And we will have meetings together on case discussion. We will review a patient's charts as well as their molecular test results, and we will discuss and come into a consensus on the best solution of the patient's treatment. And with all this genomic technology available there, we also know that translating those genomic information into clinical takes a teamwork. So we also need to spread the word so that more healthcare providers and And we need to equip our future pharmacists with the capability to understand and to utilize this genomic information in their healthcare services. So the Pharmacogenomic Society of Taiwan, we have generated several guidebooks. The one on the left is already available out there. It is mainly focused on the companion diagnosis for the anti-cancer drugs that are covered in our own national health insurance. Well, the one on the right, it is actually a guideline for applying pharmacogenomics in clinical pharmacy services. And in this book, we include not only the dosing recommendation of the drugs, we also include some counseling skills and advice on the counseling sessions. And we also have chapters talking about the requirement for the laboratory that is needed for analyzing those clinical samples and genetic analysis. So in this guidebook, we work with the Taiwan Society of Health Systems Pharmacists, and it took over 40 pharmacists around Taiwan to generate this book. And this one is still in progress, and we plan to publish it in this year, so please stay tuned, and this will be available in near future. So I think it is the end of my presentation, and I'd like to take this chance to show my acknowledgement to some people and to some people. And first of all, I'd like to give special thanks to Dr. Ellen Zhao and also Taipei Medical University, because for their support of sending me and also other oncologists and pathologists to be trained for the GCRA in City of Hope. And I also like to thank Professor Wei Chao Zhang, my PhD advisor and TMU School of Pharmacy, which led me to the field of genomics and pharmacogenomics. And I also like to thank TMU Hospital and Taipei Cancer Center. They are really supportive and active on developing those precision medicine healthcare services. And I also like to thank the partners from PGST and TSHP on generating those wonderful guidebooks. Thank you very much.
Thank you, Dr. Zhou, for the very comprehensive overview of the genomic analysis in the cancer risk assessment, diagnosis, and the treatment selection. So this is time for the audience. If you have any question for Dr. Zhou, please raise your hand.
Thank you so much for that really comprehensive overview of both the germline and the somatic genetics of tumors. I have a question. When you're performing the tumor profiling, either in solid tumor or the liquid biopsy, as you mentioned, you're going to be able to identify both germline and somatic mutations. What do you have in place in terms of pathway for clinical referral, or how do you go about following up when you suspect something might be of germline origin, get patients referred for genetic counseling and further genetic risk assessment?
Okay, that is a really, really great question. And for that, when we look into the cancer genomic profiling, the very important thing is that we always refer back to the laboratory that offering the test, because for now some of the laboratory, they will deduct the germline variants in their report, but some of them don't. So the first thing is to make sure about this point, and if there is a possible germline change in the results, we often recommend the attending physicians for further germline testing. But the other thing is that because in the current situation in Taiwan, those germline tests are not covered by insurance, so it is very difficult. We can give the advice, but most of the time, because when it comes to cancer treatment, the prioritization of where to spend the money is also very important. So normally, the patient would discuss. Normally, they will decide that they can focus more on their treatment first, and then the germline later. Yeah. Thank you.
Okay, following that question, I want to ask some practical question, including, do you think the turnaround time for the genomic profiling is fast enough to inform the treatment on time? And right now we know that there are many genetic testing laboratory, and they may use different sequencing platform. So how can we ensure the consistency, the accuracy of these different platforms to give the report?
Oh, thank you. That is a really big question. And in terms of the turnaround time, because in clinical setting, of course, we want to get the results as quick as possible. Because when we wait, actually, the tumor itself is also changing itself. Mm-hmm. It is evolving. So as far as I know, a lot of hospitals try to get those testings in-house, get the in-house testings, which may ensure a faster turnaround time. But to take the lab test in-house, it takes a lot of effort, and yeah, it needs a lot of discussion across different departments with the laboratory, the medicinal laboratory, mm-hmm. and with the pathology and the interpretation of the results. Mm-hmm. Yeah. Thank you.
Thank you.
Thank you very much. I wanted to ask, I think one of the difficulties with circulating DNA is that obviously normal cells also acquire somatic mutations, yeah. and they're not necessarily cancer associated. So do you look for a specific, is there a specific panel that you try and if you find a mutation in this gene, then it's a problem? Or do you just look for any mismatch with the background genome?
Actually, no, because some of the because in the literature we can know that some of the variants are commonly found in those somatic mutations, like the TP53. Actually, it just becomes the frequency of the variants is very high, and especially when the patients get old, there is a term called CHIP. As I say, but our human body will accumulate the genetic change naturally. So sometimes the variant you found is actually from those CHIP effect. So basically, we look into the literature and see which are more suspicious, and especially for cancer genome profiling, and we are more focused on those we can that can be treatable. We can have the target therapy on them. Yeah, thank you.
Because of time, we can only allow one more question. Okay, I want to know your opinion. In the next 5 years, what area of genomic medicine is the most likely to give the breakthrough in oncology?
I think we have a lot of discussion in the other presentations, but what I think the one I think the most promising is the one that following the path of immunotherapy, because that is really a transformative treatment option. And especially because when it comes to cancer treatment, the major question is always how our cancer tumor is different from our normal tumor. How is the cancer cells different from our normal cells? So as far as I know, that we have labs that are developing cancer vaccines, which are actually treatment vaccines for treatment, not for prevention. The treatment vaccine actually utilize the immune responses, and we can identify the cancer specific mutations and make it into the form of vaccination. When we can trigger the immune system in the patient's body to train the human body to know the pattern of the cancer cell. I think it will be a great chance for the precision medicine, and we can find a personalized profile of the patient's tumor and treat that accordingly. Yeah.
Dr. Zhou again.
Thank you, Dr. Zhou, for answering all those questions. If we could have you on stage just for a little bit as we have our moderator present you with gifts. Thank you very much. Ladies and gentlemen, we'll now take an afternoon tea break. Refreshments and snacks will be provided outside, and we will resume promptly at around 3:15 or longer, depending. So take this time, recharge, refresh yourself, and go to the toilet if you need. Thank you. Hello, everyone. Welcome back. I hope you had a very enjoyable tea break. The snacks were very delicious. Let's now welcome Professor Yiming Liu, Chair of the PhD Program in Drug Discovery and Development Industry of the College of Pharmacy, to introduce our next speaker. Let's give her a round of applause.
Good afternoon. I'm Yiming Liu, and this is my great honor to have a chance to introduce the next speaker, the Dr. Gao. He's graduated from TMU and now is an attending physician in TMU Hospital. And today, he will discuss the clinical significance of the HRD in oncology and its role in guiding personalized and precision cancer therapy. So let's give a warm welcome for his speech.
Thank you, everyone. Today, I want to talk about the HRD signature and the germline HRR gene mutation in the cancer patient. This is my outline, and this is my case. Is a elderly woman. Is 86 years old, and she initial presentation she got an epigastric pain. After the physical examination, we arranged a CT scan, and CT scan find a huge pancreatic tumor, about 5 centimeters in diameter. So we do the US biopsy for this tumor. This tumor, the pathologic report is the adenocarcinoma, and the further staging shows she have the multiple lung metastasis, or is the initial diagnosis stage 4 disease. Then, because she is very old and frail, so we cannot, she cannot tolerate the high intensity chemo regimen like for FOLFIRINOX. So we using the gemcitabine based therapy. We use the gemcitabine plus neck tarsal ablation, and she received 3 cycles of the chemotherapy. During the chemotherapy, despite of we using a very, very low dose, only half dose of the standard dose, she still have the grade 2 neutropenia and the thrombocytopenia. So my patient is not very tolerant to the chemotherapy. And the response evaluation, the tumor marker decreased from the 2,000 to 800. So by the tumor marker response is not very poor. But the CT scan show the tumor size did not decrease, only stable disease. So it's make my patients disappointment because the tumor did not have a shrinkage. And her echo status was 1 to 2, so we cannot using the high intensity chemotherapy for her. And we trace back to her family history. She got a pancreas. Oh, sorry, the word is very small. She got pancreatic cancer, and when 1 year ago, when she was 85 years old, and she has 3 brothers and 2 sisters. And 3 of her brother all dead because variant etiology. One of her brother died for stroke when 50 years old, and one of brother died due to the suicide and when he was 50 years old. But one of his younger brother died for pancreatic cancer when 78 years old, and her younger sister have have breast cancer when she was 50, and she had a contralateral breast cancer when 55. So her younger sister have multi cancer, despite all breast cancer, but the bilateral primary breast cancer. So this is a very interesting family history, and the son of her younger sister also have colorectal cancer when he was 40 years old. But my patient, her husband also have the colorectal cancer, but her son and the grandson and granddaughter do not have any cancer history. So in raising my interest about this family history, look for this pedigree. Many of her family members got cancer, but the age is not very young. Only her younger sister and her family has a young cancer history. However, according to the NCCN guideline, or if the patient got the pancreatic cancer, if one of the first degree relevant have the pancreatic cancer, it meets the criteria for the genetic test. So I arranged a germline genetic test for her, and she got a FANK-M mutation. This V1336FS mutation is a heterozygosity mutation. But it makes sense because the homozygous will cause Fanconi anemia, and according to ACMG classification, it's a likely pathologic mutation. But this variant is not a common variant for the HR gene. It's not very common. So I searched from the Canva, but no large data or no many data about this mutation related to pancreatic cancer or breast cancer. So what's our next step in current evidence? So we can review the DNA repair mechanism. Our DNA breakdown damage can divide it to the single strand breakdown and double strand breakdown, or base mismatch, or Barkey lesion cross-link. In the single strand breakdown, we're using the BR, or base excision repair, and for the mismatch, we're using a mismatch repair protein. And for the double strand breakdown, we're using the HR homologous recombination, or NHEJ, the repair system. And how the double strand breakdown and the homologous recombination repairing work. In the first step, the double strand breakdown happen. Our mRNA complex will produce a 3' OH ending tail, and this tail will binding to the replication protein A, and the replication protein A will form with the RAD 51, and this will cause a spatial arm and causing a homologous invasion into our homologous DNA, and the homologous DNA will start repair according to this arm, and it will cause three kinds of result. The first result, the DNA start repairing and get attach back the breakdown arm of the 5', then start the repairing. So it's showing a no cross reaction repair. It's called synthesis depending stranding annealing. And another condition is break-induced replication. It means the replication will occur to the homologous arm of the DNA, and the repair will according to the homologous arm. So it will cause the loss of heterozygosity because the repair arm is same with the another homologous arm. So the heterozygous will lose it. And another condition is leading the crossover, because our homologous arm, then the other part of DNA will binding to the division arm, and the division arm will cause a crossover by the cutting end of the DNA fork. So we can see the blue bar and the red bar will change its ordinary DNA site. So it causes a crossover reaction. It's a double strand break repair. So it's a very complicated for HR repairing. During this repair, using the very complicated repairing, it can largely decrease the repair mistake during the repairing process. And we also mentioned about another non-homologous ending joint repairing. This kind of repairing is the 3' OH end forming, and it will delay causing the delayed repairing from the resting of the DNA. So it will cause DNA deletion after the repairing. It will cause some kind of a genomic scar during the repairing process. So the homologous recombination repairing will not have this phenomenon. It will cause some spatial phenomenon in the homologous recombination deficient patient. Okay. The Frank complex is quite very complicated because the Frank family have many kind of protein, like the most family like Frank A or Frank B. It's a common mutation in the Frank-related gene mutation, but it still has other like Frank F, Frank C, Frank N. For example, our patient is Frank M. And in the repairing process, every protein have its unique function. Like ROD51 is binding to the DNA, and the most famous part of it is BRCA. The BRCA2 will direct binding to the ROD51. So the BRCA A and BRCA1 and BRCA2 is a key protein in the HRD signature, but other parts still have an important function. So by the definition of the HRR-related gene, it has it's a very complex, like DNA recognition and the repairing protein, BRCA1, BRCA2, ROD51, PLB2, ATM, CHEK1, CHEK2, and family. So did every gene have the same effect on the repairing system? I think it's no, because the BRCA, BRCA1, BRCA2 is the most important key protein, and the ROD51 is also a key protein in the repairing system. And how we detection to the HRD signature? Because as above mentioned, the HRD signature is related to the HR gene mutation. So we can using the direct HR gene mutation analysis. However, the genotype is not means the phenotype, because some mutation is not the pathological mutation. So we can in most commercial panel, it will add on another HRD signature like a genomic scar. The loss of heterozygosity and large-scale transition, thermic allele imbalance. This condition will happen if the HR is deficient condition. The repairing cannot do this kind of repair, so it will cause this some DNA defect in the patient's gene. So it called a genomic scar process. We can direct detection the genomic scar to find out the HRD signature. Another is the mutation signature, like the most famous is signature 3. Signature 3 is an indel mutation. It's most relation to BRCA gene. And another is functional assessment. Like, for example, we can direct test the ROD51 analysis for its function of the HR repairing system. So it's a comparison. Sorry, it's still some Chinese in this table. Like above mentioned, we can test the signal 3, loss of heterozygosity and large-scale transition, and the thermic allele imbalance, and we also can, using above 3, can calculation an HRD score. And this score in many commercial kit can using to test the HRD signature. Like this table, for example, the foundation medicine, I always using this panel. In the foundation panel, they using the loss of zygosity plus the BRCA gene as the HRD signature detection. So it's a one way for the HRD signature detection. And another, like the Harvard Medicine School, is a Sigma system, using the more like a next generation sequences to sequence more than 300 genes for the HR gene mutation, and it also tests the signature tree and the loss of zygosity, the scoring system. And another, like we can use in a whole genomic system or whole exome sequencing for detection the gene. So it's many used for commercial assay for detection the HRD signature. And how the HRD influence our cancer immunity, just like the microsatellite instability condition, the HRD signature will cause the tumor mutation burden will increase. So in the Intel mutation will increase the tumor mutation burden. It also will cause the cancer neoantigen. In the condition with many cancer neoantigens, it will cause it will induce the checkpoint signal elevation. But however, on the other hand, in the HRD signature, it also causes the copy number variation. In the genomic scoring system, it will change the copy number variation. In copy number variation increase condition, our T cell function will reduce, especially CD8 positive T cell. In the CD8 positive T cell is most important in the tumor infiltration T cell. So it also inhibition the cancer immunity. And additional, the stick signal will enhanced by the DNA damage. After HRD signature HR deficiency, the DNA damage will increase, and the DNA damage increase will activation our stick signal, and signal will increase our interferon pathway, like interferon alpha and the CD8 positive T cell. So the HRD for the cancer immunity is heterogeneous, or it will sometimes increase our immunity by the checkpoint pathway. On the other hand, it will inhibition our immunity through the copy number variation and the T cell cytotoxicity. So for the HRD positive cancer, we should using a checkpoint inhibitor or not is still a lack of a large phase 2 or phase 3 study for the treatment outcome evaluation. For example, like in the breast cancer, the BRCA1 mutation will increase the TO function. So in the BRCA1 mutation, or like a triple negative breast cancer, the checkpoint inhibitor may be effective in this situation. And for the ovarian cancer, is also the TL and the PD-L1 expression will increase in the HRD positive patient. In the prostate cancer, some studies reveal the BRCA2 mutation will increase the TL number than the non-tumor part tissue. So it's some histology evidence to show the immunity and the HRD signature, the relationship, and it will also change our practice for the cancer treatment. Another part is for the chemotherapy. As above mentioned, the HR mutation is related to the double-strand break injury repairing. So we all know the platinum-based chemotherapy and the topoisomerase inhibitor is very effective to cause the double-stranding breaking injury. So we know the cancer with HRD signal is sensitive to the platinum. However, how we choose our treatment, because we can use a platinum-based chemotherapy or using the PARP inhibitor. Which kind of agent should we use as the first-line therapy? So it's very difficult to determine. However, it's very much evidence to debate about the platinum-based and non-platinum-based therapy. For example, in the pancreatic cancer, this is a paper discussion about if the pancreatic cancer have a HRD signature. The fulvestrant is more effective than the gemcitabine and paclitaxel-based therapy. We know the fulvestrant have the component of the oxaliplatin is one of the platinum, and the irinotecan. Irinotecan is a topoisomerase 2 inhibitor. So the fulvestrant in many phase 3 data, fulvestrant have more prominent progression-free survival and overall survival. I think maybe one part of the HRD signal possible pancreatic cancer have the attribution to this survival benefit. In this study, it's a retrospective study and include about 8,358 patients. And the HRD signature positive is accounting 9% of these patients. For the outcome, this is overall survival. This is the survival, the time to next treatment. So we can take it as the progression-free survival. It's similar to the progression survival. And for the HRD positive, the red line is the HRD signature positive using a fulvestrant. Both of these, two survival lines show the HRD positive patient using the fulvestrant, the platinum-based plus topoisomerase inhibitor can have the better outcome. So it's evidence for how we choose our chemotherapy in this kind of patient. Another trial is the TNT trial, is a triple negative breast cancer. It's a phase 3 prospective randomized control trial. It includes a patient with triple negative breast cancer and with BRCA mutation. Enrolled 378 patients and randomization to two arms. First arm is carboplatin based, and another arm is the docetaxel based. The primary endpoint is overall response, and the second endpoint is progression-free survival and the overall survival. And in the overall response evaluation, the all population, these two agents similar response. But for the germline BRCA mutation and the somatic BRCA mutation, we can see obviously the carboplatin-based in the mutation patient, the carboplatin-based chemotherapy have a better overall response, and the p-value have a significant in the two kind of difference between the two kind of therapy. But in the wild type patient, the two kinds of therapy did not have a difference and have the trend made better in the docetaxel arm. And in the sporadic condition, the tumor have only tumor have a BRCA mutation. It still have the significant response in the carboplatin arm. However, the p-value did not have the statistical significance, so we cannot conclude these two kind of regimen is effective because the n number is too small. The only five patients have a BRCA mutation in the sporadic condition, so may have some problem in the power. Right? And same to the germline mutation, the wild type did not show the overall response benefit in the carboplatin-based therapy. And the response can translate to the overall survival or progression-free survival benefit. The answer is yes. We can see if the patient have the whether germline or two sporadic mutation. If the patient have a BRCA mutation, the survival outcome is this orange line, or it's better than whether you using the docetaxel in unmutated or mutated patient. However, if the BRCA is wild type, you using the platinum base, the survival is the worst in these four subgroup. And same in the sporadic condition, the overall survival we can see is the best in the BRCA mutation patient using the platinum-based therapy. So in this phase 3 study, we can conclude the platinum-based therapy is better in the BRCA mutation, especially in the germline condition. But on the other hand, if the patient did not have the BRCA mutation, you have better choosing a traditional like a taxane-based or anthracycline-based therapy for this kind of patient using the adjuvant chemotherapy. Interestingly, for other HRD marker, this journal using the HR deficiency status like HRD score and the basal-like signature. We all know the basal-like signature is according to the microarray result, and is similar to the triple-negative patient's result. Or you using a traditional IHC pattern, the triple-negative pattern. In these four conditions, other HRD marker do not have the benefit in the platinum-based chemotherapy. And besides, in this condition, if patient have a basal-like signature, the taxane-based have a better response than the platinum-based. So for other kind of HRD signature, it seems the chemotherapy is not very reliable other mutation than the BRCA mutation. Okay. Then we talk about the PrEP inhibitor for the whole HR mutation cancer. We all know the PrEP inhibitor, the mechanism of PrEP inhibitor is due to if the patient have normal repair, the PrEP inhibitor can binding to a single break, and a PrEP will binding to a single break and enhance the DNA repairing. But in the HR mutation patient, if it got a double strand break, you using the PrEP inhibitor, the PrEP cannot healing the single strand break. It will cause a double strand break, and the double strand break cannot be repaired by the HR family. So the cell will have the enhance the DNA break will increase and leading the cell death. So it's a mechanism for PrEP inhibitor and how it works. And currently we have the 6th kind of the PrEP inhibitor, but in Taiwan we only have the olaparib is available in our health insurance system. So many kind of patients, despite some evidence is using other kind of drug, but we only can using the olaparib for treatment. However, there's not many evidence in the other than BRCA mutation HRD signature using the PrEP inhibitor because it lack of sufficient phase 2 or phase 3 study in this condition. This is a real world data for the ovarian cancer with BRCA mutation and the other HRD signature. The total include 673 patients. It's a retrospective data, and the treatment as a monotherapy maintenance after platinum-based chemotherapy. And of the 600 patients, only 116 patients under PrEP inhibitor. And the definition of the HRD signature is mutation of BRCA1, BRCA2, ATM, ROD1, PrEP1, CHEK1, CHEK2, and FANK-LO. probably 2, right 51B, right 51C, right 51D. All this kind of mutation is defined as the HRD signature in this journal. And we can see if the patient have the BRCA mutation, the progression-free survival is obviously longer and have the long plateau. It means the patient will get no recurrence under the maintenance therapy. The progression-free survival is 21 months at 21 months. Median progression-free survival is 21 months. In wild-type patient, you can see the survival curve is the same between the no maintenance and using the PrEP inhibitor. And for the progression for survival and the overall survival have the same trend in the BRCA, BRCA mutation and BRCA wild type. And for other HRD signature positive, the trend is more obviously in HRD signature positive patient using the PrEP inhibitor. We can see the progression-free survival and the overall survival is better than BRCA mutation only. So these real-world data support we using the PrEP inhibitor in the situation of other kind of the HRD signature positive the cancer patient in the ovarian cancer. However, we still not have sufficient data because no prospective study. So I introduced one journal about the phase 2 study. It's for the talazoparib monotherapy after patient have the HRD signature other than BRCA1 and BRCA2. In this study, it includes 20 patients and 13 is breast cancer and 7 patients other type of solid tumor. All have the HRD positive mutation other than BRCA mutation. It's a whole data analysis. We can see other mutation including ATM, ATR, and PROB2, CHEK2, FRANK8, and P10, and RED50. And not all patients germline mutation. Only some patients, still 9 patients have somatic mutation, and some patients have both germline and somatic mutation. The primary outcomes is overall response. We can see only breast cancer have the excellent response. 31% have partial response, and 46% have a stable response, but still 20% have progressive disease. In non-breast setting, no patient have tumor regression, and only about 57% have a stable disease, and 43 have progressive disease. So in overall response, about 20% have tumor regression, and health patients have stable disease, and 30% have progressive disease. The ORR is about 20%, and the disease control rate is only 55%. So obviously the data is not, we're not satisfied in this situation, because in the other phase 3 study with a BRCA mutation, the PrEP inhibitor have a better outcome than this data. And we can further analysis different kind of HR gene we can see the waterfall data. The PROB2, most of the responder is PROB2 mutation. And for the duration of response, it's heterogeneous. Like the first patient have a CHEK2, FRANK8, and P10, and the second patient is CHEK2, and then the third patient PROB2, P10. So if the patient is responder, the duration of response is not a correlation to the genetic profile, but response rate is highly correlation to the specific mutation. In the regression model, we can see the probability to have the best response in the other than BRCA mutation, HR gene mutation cancer. So we can take back to in every kind of cancer. In the SOLO1 study, PRIMA study, and the PALA study, the SOLO1 study is for germline BRCA mutation. The PARP inhibitor have the progression-free survival benefit. And in the PRIMA study for germline BRCA and the HRD gene, both HRD positive patients have a progression-free survival benefit. In the POLA A study, the HRD positive patients have the progression-free survival and the overall survival. And in the acceptable monotherapy, the BRCA plus the loss of zygosity HRD signal, it also showed progression-free survival benefit. So in the ovarian cancer, if the patient have the HRD signature other than BRCA mutation, it seems the PARP inhibitor still have the progression-free survival benefit. In breast cancer, most study is focused on the BRCA mutation. So maybe only BRCA mutation have the progression-free survival benefit after the metastatic setting. In the prostate cancer, the BRCA mutation is whether germline or somatic. The HRD marker in the PROFOUND study is using the BRCA and ATM. Both have the progression-free survival. And the TRITON study, the germline or sporadic BRCA mutation have the response and the overall survival study. And in the Galhead study, the BRCA and the non-BRCA, in this comparison, the PARP inhibitor only have the most benefit is in the BRCA mutation patient. But in the non-BRCA mutation patient, the ATM that check to have the limited response to the PARP inhibitor. So in pancreatic cancer, only the germline BRCA mutation have benefit in the progression-free survival. So summary above phase 2, phase 3 study in the non-BRCA mutation HRR, only ovarian cancer have sufficient evidence to support our using the PARP inhibitor in this condition. And other cancer type did not support we using the PARP inhibitor in this situation. But chemotherapy can support in the pancreatic cancer with using the platinum-based in non-BRCA mutation patient. So this is a summary of ISMO guidelines. For ISMO guidelines, whether germline and the somatic mutation of the BRCA is a good marker for predict the HRD signature and using the maintenance therapy. But however, for the non-BRCA mutation, there's no sufficient evidence, so it must take it case by case or determine the specific mutation for the HR gene. So back to my case. However, we don't have the sufficient evidence. You suppose we're using a PARP inhibitor, but this patient have no other available drug because she cannot tolerance the fulfillment. So we still try the olaparib, one standard dose olaparib. Fortunately, the tumor marker have a further regression. So her tumor marker in this month is only 472, and the initial tumor marker is 2,000. But the tumor response is still shows stationary in the primary site and the lung metastasis. So I think the PrEP inhibitor in the other than BRCA mutation in breast pancreatic cancer, it truly did not have the a standard response, because back to the Frank M, the function of Frank M, the Frank M, it seems not a key. It's only it's like just an adjuvant role in this HR repair system, not like BRCA1, BRCA2, and ROD51 is a key component protein. So I think despite you inhibition this protein function, it do not make this whole repair system have efficient DNA damage to leading the cell death. So it can only cause the disease a stable condition. But however, we still lack of a large prospective study to support our using the PrEP inhibitor or platinum-based chemotherapy in the other than BRCA HR gene mutation. It's my presentation. Thank you.
Thanks for the impressive talk from the Dr. Gao. And now we are opening for the questions.
Thank you very much. I believe there was a slide where you showed that the HRD patients had better response to PARP inhibitors, or was it PARP inhibitors? I now can't remember, but they had much better progression-free survival, and I think the overall survival was bordering on significance, but the difference seemed to be smaller. And so I was wondering if you had any ideas why that might be happening. Why is it that they progress much slower, but then overall their survival is not necessarily that different?
I think maybe one of the reasons because this kind of the patient is due to a germline mutation, so maybe the patient will get the second primary or third cancer. So despite this cancer will decrease the tumor burden, but if another hit will cause you cannot prohibition another cancer formation. And another for drug resistance mechanism is from the BRCA methylation, because in some cell model we can observation if you using a PrEP inhibitor, you will cause the methylation for the BRCA protein, BRCA gene. If the BRCA gene have a methylation, will knock down. If knock down the HR deficiency, will become the proficient status. So it's another way to explain the drug resistance mechanism. I think it maybe a drug resistance cause the cancer will further progression after drug resistance formation.
Thank you. I think one of the recurrent themes that bothers me that comes up over and over again is the discordance in results between tumors which are HRD positive, genomic scar, versus either somatic or germline. You know, different tumor types seem to respond better in some categories and not others. And I wondered if you had any thoughts. Do any of the studies that you presented, for example, look at maybe zygosity? Is there actually a second hit in the tumor? There may be, you know, non-BRCA related tumors happening in germline carriers to explain why some germline carriers, for example, don't respond the way we would expect, or vice versa. What do you think accounts for that discordance that we see in different tumor types?
It's a very difficult question. I think because not similar to the Lynch syndrome. The Lynch syndrome only the small group of for the mismatch repair protein, but in the HRD, many protein is form this pathway in the repairing system. So we do not know actually every protein the importance of each protein. So we do not have the. I think it maybe should have one scoring system for the phenotype, because we don't know the mutation, because we do not know the mutation, how the contribution to the repair system. Maybe it only have the divisions of the mutations, this repair system only 10%, or another mutation will cause more than 90%. And further, in the different kind of cancer, the oncogenesis I think is quite different. So maybe additional mutation is very important. Just like we have the strong driver mutation. This kind of HRD maybe is one of the strong driver mutation, but maybe other additional mutation will influence the HRD signature itself. So I think it's very complex to determine how why different cancer type or why the response. And I think we still have some prospective study to support our theory, or a further cell model to support our theory. But I think it's very difficult to perform this study because of different cancer type and different adjuvant chemotherapy and different mutation signature. So I think before we perform this kind of study, maybe we have the standard scoring system for the whole HRD gene, HRR gene, to calculation one scoring system to determine every gene, like a PIS score for the multi-gene, the result of the multi-gene. I think maybe this next step research to how we can do.
Any questions? Okay, so for the time limit, okay, so thanks to the Dr. Gao again. Thank you.
Thank you.
Thank you, Dr. Gao. If you could stay on stage, and we'll have our moderator present a certificate and gift for your visit and contributions. Thank you. Thank you very much. Now, to moderate our final presentation for today, we will invite Professor Jiarong Li, Chair of the Graduate Institute of Pharmacognosy and the PhD Program in Clinical Drug Development of Herbal Medicine. Let's give her a very warm welcome.
Okay, thanks all for you to stay here till the end. So as we know, the last is the best, right? Okay, so today we are so glad to invite Dr. Chen to come to here. And Dr. Chen graduated from National Taiwan University and got his degree in the University of Missouri in pharmacy and pharmacology. And she has a long experience work in FDA for more than 25 years. And after retiring, Dr. Chen established pharmacovigilance consulting in the U.S. And she provided the expertise in regulatory and clinical safety consulting in area of the product development and the pre and post-marketing clinical safety to the pharmaceutical industry, regulatory authorities, and the training of the academic organization and the industry. So, as we know, the pharmaceutical education safety is the first steps. Okay, welcome, Dr. Chen.
Well, what time is it now? Good afternoon. I'm the last one, but as she said, could be the best one, who knows? But I really want to congratulate Dr. Alan Chow. Where are you? I want to give hats off to his really sponsorship to this conference. Really, really wonderful. And then also for the organization in the Taipei Medical University and all the staffs putting together such a wonderful conference. This conference gathered all the people around the world. I'm from US. I think Dr. Hoke is from US, and then Dr. Somera also, and then from UK. So we are very lucky to be able to gather together here to share all our experiences. And then for all the attendees, you're lucky to see so many people here to give us the most advanced pharmacogenomic information or research. I was just overwhelmed. I don't know about you, but I was overwhelmed. So from now, I think really I would just, everybody can really relax. There's no gene mapping here, okay? You can just sit there, relax, and then listen to my stories. I like to say these are my stories on my the journey in FDA, working as a director, finally as a director of pharmacovigilance. So it's a very interesting experience, and then very relevant to the pharmacy practice, and also very relevant to the drug discovery in pharmaceutical science area. You don't think it's relevant? By the time you finish my talk, listening to my talk, you know it's very, very important as far as drug safety in drug development actually can assure you the drug can be developed safely for human use. Okay? So I don't think I have said that earlier, but you know what? Let's give everyone a big round of applause, to yourself, to the people, speakers, and the workers here. Okay. I'm very fortunate to be the last one, so I like to wrap up a little bit in these 2 days, all the talks. It's full agenda for 2 days. I was not expecting this, so much information. So what I have felt from this conference is that you have put together a very good speakers from different places to give us a general overview of the pharmacy education. 4 years, 6 years are very similar in different places. I love Dr. Hoke's the King's presentation that give everybody really the overview of everything. And then Dr. Samara gave us the clinical setting in oncology centers in City of Hope. City of Hope, then give you a chance to know how she operates the pharmacy system in the United States in oncology area. Then we have all the different people talking about, a series of people talking about the research ideas in genomics, including Dr. Okada. I don't know where you're here, but I do salute to you as a genius of this, right? And a lot of research ideas, and I couldn't get half of it, okay, or a little bit. Then Dr. Asai talked about the pharmacognosy, biosynthesis cluster discovery, which I really didn't think that was so advanced, because to me, I learned the pharmacognosy in National Taiwan University, but once I got to the US pharmacy school, they don't teach this, okay? So I said, we are revisiting this again? Wow, no wonder. We're talking, he was talking about actually a different synthetic process for this nature product, which really is eye-opening. You'll save a lot of production or manufacturing cost by using this biologic process. So I really appreciate his input here. Then we have Dr. Fairfax from London talking about that, what do you call that thing? It's something is checkpoint. Yeah, okay. So this is very eye-opening, and then you actually talk about adverse event. Am I right? Yeah. So that is to my heart. I'm glad you touched it. Thank you so much for all the information. And then we have Dr. Zhou and Dr. Gao and Dr. Wu talking about some reality check in clinical setting, how we can treat patients with different options when they have mutations, gene mutations, and a different kind of cancer in this research. And I think this is very, very important practice options for the physicians here and worldwide to think about what kind of study they can use to enhance their therapy treatment options for patients they actually take care of. And then the most intriguing presentation was from Dr. Gross. You remember him? He is not here, but he is from Illinois, Chicago area. What he did is presenting some of the study information as a clinical pharmacist that looking at the kidney damage from different drugs, which I was very, very impressed, because that small studies has pros and cons, and that's what happened to the real world. There are many, many clinical pharmacists on their job. They actually love to do studies. The studies are small. You can't create any significant differences in comparison. However, they will present the information and then let people to decide whether that's worth using or not. So that's what I have learned from this conference. Do you agree? Anyway, so I'm glad to see you are staying here all the way to the end. Okay. So first thing is I want to talk a little bit about my professional journey, because a lot of people wondering, why did you get into drug safety area as a pharmacist, right? In the FDA. So it's a non-orthodox pathway. However, it works for me. So I really like to share what happened. I graduated from National Taiwan University not as a good student. So not as a good student, but I didn't try to catch with all my classmates going to the USA to get graduate degree, because I was too late to apply. So I have to go to graduate school and say, look at my records, college records. Say, which graduate school I should go, you know? It takes exam. It's not like in the US you apply, but here you have to take the exam to go to graduate school. So I look at the records and whoops, pharmacology is the only one that can show that I'm better than other areas. So I went there and studied the pharmacology. Who knows? I went to the USA, study pharmacology again. However, I got married, I have children, and my husband is also a pharmacologist. So both of us, like staying together, working in the same city is almost impossible. So I decided to give up my study, and then went back to get my license. Went to school to get my get a BS degree. At that time, they didn't accept the accept the Taiwan degree, BS degree, pharmacy. So I have to go back to school there to get my BS degree and take the exam to become a registered pharmacist. And luckily, University of Missouri back then was a very, very clinically oriented pharmacy school. And later on, they did have this program, I think a US famous program, and I was lucky to get some training there. Actually, I was qualified to study the medicine when I graduated from high school. However, it turned out I found out I cannot stand the bloody things. Just like Dr. Samara, she said she couldn't be a dentist, right? So I couldn't be a doctor. So I decided to go into the chemical world, pharmacy. But you know what? I didn't like to be a dispensing pharmacist either. So what should I do? So I went to graduate school. But once I graduated from pharmacy school, University of Missouri, I had to work to make money. And at that time, with children, 2 young children, what can I do? So I decided to work part-time. So that's the benefits of being a pharmacist in the U.S. You can work part-time in a retail pharmacy and work in the Rite Aid in New Jersey, okay, from 3 o'clock to like 9 o'clock. And it was turned out a very, very interesting experience. That experience actually opened up my mind, what I can do for patients when they walk into the pharmacy. For example, one time there was an old man coming into the pharmacy and gave me a bottle. He said, I want to get this refilled. I said, okay, you have to wait for 15 minutes because I have other people here ahead of you. So what? All you have to do is just pour the medicine from the shelf to my bottle. I have to wait for 15 minutes? I said, yes, there are people ahead of you. Besides, I want to know what you're taking. And I want to talk to you about the medications you're taking and the possible side effects you may encounter. He couldn't wait. He grabbed the bottle and went away. Okay, you know what? Next day, he came back. So I patiently talked to him about all the medication issues with him. He went away happily with smiles. So this is something at first I realized, yes, I can do patient care this way using my knowledge and then also with some passion, and that's a very important thing. So, but that's for a short time. I went to the medical center to work, and then eventually followed my husband to FDA, but at that time I was working at NIH Clinical Center. Now, NIH is a very different, unique place, a pharmacy, because NIH is an institute that does all kinds of investigational drug. So they actually make investigational drug on site and then dispense to the patients that need it. Those drugs are not approved yet, all investigational under the IND. So the patients, they have no other treatment. They have to go through this process in order to get some new treatments available at that time by NIH. So it's a very, very unique place to work, but every day you're dealing with investigational drugs, and then the patient very, very sick. And what I really admire the place is that NIH offer a McDonald house right across from NIH. The patient family can stay there for free while the family member is being treated there. So that was quite an experience. However, I quickly went to US FDA. I just found out from this conference, Dr. Samara was in NIH. Okay, it's funny how we know each other that way. And then I was there first. I think she later came. So it's very interesting that it's a famous place to work there. And then I went to FDA, referred by my husband because he was there first. I said, what does FDA do? Do you know what does FDA do if you're outside, you're practicing dispensing? Nobody really knows that much at all. All I knew was that long prescribing leaflet, right? And then you don't even see it most of the time. So what does FDA do really to offer this long list of prescribing information to the doctors or maybe to the patients? I had no idea. But before I knew everything, I loved the place. I learned everything about drug approval, learned everything about drug safety. So this is the place that actually I found out really this is something satisfying my professional skills as pharmacologist, and also serve my not actually ambition, but somehow the professionalism in this area really got me promoted. So this is something that I want to say, I didn't know what happened, but it's naturally going there, and this is probably a best experience I have in my life. So to start with the drug safety, I'd like to ask you a question. Have you heard about Vioxx? The new people, young people, you probably never heard about. But if you are 10 or 20 years older than than a new graduate, you probably have heard of Vioxx, very famous drug that got pulled from the market in 2004. Why? What happened? So I'll explain that a little bit. Medical products are getting more and more complex and going global for small molecules, biologics, cellular therapy, gene therapy, vaccines, and devices. You have heard a lot in these 2 days. So the goal for medical care now is to ensure that the right product is delivered to the right patient at the right time, right? You agree? That is called precision medicine. Are we doing it now? Yes, we are. With all the new inventions, drug discovery, biologics, gene therapy, we are doing that right now. However, we can't do it on our own. We have to be regulated by all the regulatory authority in different countries. So they are the guardian angels to make sure the product is developed correctly and safe and effective for patient use. So when you develop new drugs, you got to have some innovative ideas in research, and then also to study the target condition, available treatment for the disease you're trying to reach. And then also after clinical trials, you have to assess the benefits and risks to see whether actually patient can benefit from this, and then also the risk can be managed in the life cycle of the product. So the drug has to be going through the preclinical testing in animal, and then clinical phase 1, 2, 3 for final submission to the regulatory authority for approval. As a corporate management or investors these days, you have to make some decisions. These decisions at different stages, the preclinical testing, IND phase 1, 2, 3, or NDA filing time. All these stages can yank your product for safety reasons or efficacy problems. Okay, so this is a big investment. You have to be careful what you're doing. However, this will not kill all the scientists' dream. They continue to explore the new ideas, the new ways to develop new drugs. So this is human nature. My husband is one of them. Dreaming, now is still dreaming, trying to develop some new drugs. So just give you some idea, these dreams and innovation in drug development. This is a 2-year period of time statistics. There were about 12,000 ideas or products should be trying to be developed. Okay? But not all of them can end up in the market. Okay, so there may be phase 1, you have only this much left. Phase 2, phase 3, and pre-regulation or registration are launched. So from here to here, it's very sad, right? But you know what? This part will keep growing. Look at today and yesterday. All the genomic, the product in everyone's dream or research lab, everywhere, university, there is no stop. This is just being there, just every year. I have more numbers like this every year. So that's very, very encouraging that scientists have ideas. They want to continue developing new drugs for patient care, for our own use. So even though it's only a little bit left in the end, that is the goal, to give patients all the options they have to get treatment. So we know safety can be derived from different phases. Preclinical, phase 1, 2, 3, even post-marketing. So the safety concerns can be put together for assessment and then develop strategies and actions to minimize the risk. So we all know that. What's the approvability for marketing? It's actually all the regulatory agency, they all just require you have enough substantial evidence to prove the drug is safe and effective. Okay. What kind of substantial evidences, as you have heard about the randomized clinical trials? In the past, there were 2 required, and now could be 1, plus some supportive information supporting the efficacy. And then the evidence, strength of the evidence on the benefits and the risks, actually, you can calculate and then you can balance it. Then the drug can be approved. Okay. The risk is actually easy to describe. The risk is when people are willing to tolerate and manage it. What does that mean? That if you have a terminally ill disease like cancer, and then you have immunosuppressant agent used to have all these immunosuppressive problems like your blood deficiency, and then you get some other disease from this. Are you going to take this anti-cancer drug, even though you experience this? You may make a decision. You want to take the medications to deal with the risk separately because you are trying to get the best benefits out of this drug to survive, to live longer. You know, the cancer drugs in the FDA, they usually approve that in about a year or so time. But for cancer drugs, it's faster and faster. It can be 6 months. There was one time, it was 3 months, it got approved. You know, there is a need. Unmet medical needs. You need to have this option available in the market for patient use. So it's flexible as long as you can tolerate the risk. There's a way to get the drug on the market. So benefit risk evaluation has to be evaluated by sponsor, company, and the regulator. Also the healthcare provider, physicians, to see whether this is good for the patient use. Also for the patient, do I want to take the medications for the benefit of it, or do I want to take the risk? So every level of the decision making is a benefit risk assessment principle. However, not every drug has been approved is safe. So in the 1990s, there's a famous person called Thomas More. He published the book, Deadly Medicine, because he saw so many drugs have been killing, making patients die for one reason or the other. So we have to be careful. When things happen, what do? We have to be careful. And then in the 10 or 11 year period time, there were almost 20 drugs. When I was there, we just had a lot of, we are very busy taking the drugs off the market left and right. Okay, and Vioxx was here. Okay, so you probably recognize some of the products here. We have used this for a long time. Why they're not safe? But all of a sudden, we actually reviewed the safety profile of the market drug and found out it's not worth it having them on the market when they have so many serious side effects that causing people to die or have serious adverse reactions when there are other therapies available. It's the key. Okay, so what's the lesson learned in the U.S.? These days, you don't see a lot of withdrawals now. Okay, so what a lesson learned? The first lesson is very important. You all have heard thalidomide, right? In 1962, it's a new sleeping pill approved in Europe and found to have caused birth defects or called phocomelia in babies born in Western Europe. So they approved this drug. So the pregnant women took this for sleeping and had babies, phocomelia. And when they tried to apply this medication for approval in the US, Dr. Francis Kelsey in the FDA stopped it because she didn't think it's worth it. And this is not worth it, having pregnant women having such a big risk just for sleeping, right? So at that time, it was introduced a very important Kefauver-Harris drug amendments, starting of drug safety regulations. All the drug companies, they have to report all the adverse reaction to the FDA if they have received the information. So you think thalidomide is done, causing such a big major side effects, it's just no way it'll come back. You know what? These days, it came back. You know what? Using a stringent risk management program to restrict the use of the thalidomide in multiple myeloma, leprosy, and other indications that restrict childbearing women taking this. So you have to go, if you're a woman, you have to go through at least 2 pregnancy testings before you can take this. And after you take the drug, you still have to go through the testing again to make sure you don't get into that scenario that created an unnecessary burden on you. So that is something interesting. Withdrawn from the market or not approved the market, but it can come back. So these days, when you don't have a lot of drug available to be developed, you can revisit old drug and come back. And turned out this thalidomide is very, very effective in many other cancer treatments or severe diseases. So what happened to the 2004 Vioxx withdrawal? Okay, it was the first COX-2 inhibitor for anti-inflammatory. It's not for serious disease. Okay, and then there were some serious cardiovascular effects that patient died from MI or stroke in studies they're doing for Vioxx in multiple myeloma patients. That patient died because in this study you have an exposure denominator, you can calculate the incidence rate of the MI or stroke and find out the rate is too high. It's not worth it. So finally decided to take this off the market. It was a big, big news. And then Merck has has to go through a lot of lawsuits. You know, USA is a lawyer's paradise. They sue everything. Okay, so they have to endure these lawsuits in Merck, just like other drug products being withdrawn from the market. You get class action lawsuit too. And the other lesson learned also is the 2020 COVID-19 vaccines. As you remember, the drug trials, vaccine trials has been conducted in the US and worldwide, different countries, different populations, almost 1 million patients that showed efficacy, 90% efficacy, and then with safety profile acceptable. So in 10 months, remember I said before, it's a 12-month thing to get at least to get approval being reviewed, and then not to mention about the length of doing the clinical trials. But this, including the clinical trial and the review, it was 10 months altogether. It was approved under the EUA, Emergency Use Authorization Act. I'm sure Taiwan has it too. Almost every country has it too. But the only requirement is to make sure the vaccine is safe in post-marketing surveillance. So company has to do a very stringent and close monitoring of the safety of the patients. And you know the story, Pfizer. You know how many people got vaccinated worldwide during those years? If not 1 billion, I would say tens of millions, right? When you have such a big exposure, anything will come up. Anything will come up. So it's very hard to decide which one is actually directly related to the vaccine. We don't know. So Pfizer, Moderna has been really attacked about so many adverse events. However, it's, again, benefit and risk balance. Do you want to take it? At that time, you have no choices. You all have to take it. So this is lesson learned in these days. You don't have a lot of drug product withdrawal at all. It's good news. So what is drug safety surveillance? We start with the pharmacist on the counter, dispensing counter there. So we are usually the last line of defense in preventing adverse drug events. So what exactly is the pharmacovigilance, the word? This word is a very interesting word. It's actually derived from a French word, pharmacovigilante. So we call that, when I was there, we call our division pharmacovigilance division. It's ever since everywhere it started using the pharmacovigilance word. I like the word, so we decided to use the word. It really said that it's a surveillance process and practices to ensuring approved market medications are used appropriately and minimizing the risk of side effects, including medication errors. There are a lot of things going on in the hospital that generate medication errors and drug interaction. So US FDA defines it's the science and activities related to detection, assessment, understanding, prevention of adverse effect, and any other medicine or vaccine-related problems. So what is adverse event? You probably don't know the exact definition. It's an FDA official definition. Any undesirable, I shouldn't have it, undesirable experience associated with use of a product. So it can be from mild to severe, and it could be serious. What does that mean by serious? Serious means that they cause the event, adverse event, caused disability or life-threatening, and they result in hospitalization or death or even birth defect. These are all definition criteria for seriousness in the FDA. And what is unexpected, as we all know, remember that long prescribing information leaflet? Whatever listed there is expected. They have seen that in the clinical trial. Unexpected, they didn't see it, but now I have received reports that a patient experienced this kind of new event that never been found before. This unexpected, then you have to report to the regulatory authority for further assessment. So pharmacovigilance components: detection, reporting, case evaluation, risk assessment, and regulatory action eventually if needed. The reason I want to spell this out is because pharmacists has a big role in this practice, pharmacovigilance. So we like to identify safety signals. It's very hard. Okay, we nowadays FDA has probably over 10 or 20 million case reports in our database, right? Have you seen such a big database on adverse event? Probably not. So how can we, my department has 60 people back then, how do we use the 60 people to evaluate these cases, identify safety signals? Impossible. So what do we do? The keyword, AI. No, at that time we didn't have AI. Back then we called data mining. Have you heard? Data mining, we have a map, okay, from the collected database, the different map. The map has like a green spot and hot spot. If it's a hot spot, that means that's rare and also very serious. Then we have to take a look, closer look of that event and the drug. So it's hard. It's not easy, but we still have to do our job. It's very hard to find it. Well, these days, AI. When I left in 2014, I have established a simplified AI system for use because we have a database. We can ask the contractor just develop some kind of AI algorithm to identify the drug of your interest and identify the events of your interest, to screen the database, screen the literature, come back into our inbox. And then I'll just have to review those most serious ones as my priority of the work is assessment. So it can be done. I want to mention about case evaluation. This is what the clinical pharmacist can do. All right, we use clinical knowledge to find all the cases and then follow up with a reporter, establish the temporal relationship between the product and the event, identify the comorbidities and the underlying diseases, risk factors, and the role of concomitant meds. I think yesterday Dr. Groves mentioned about some of the factors influencing his study. Small study, but it's a study. And then we see if that's a biological possibility or it's predictable or idiosyncratic. So this whole thing, case evaluation, is perfect for pharmacists, clinical pharmacists' job. And I want to mention, back then, I have medical doctors, clinical pharmacists in my division. That when we hire people, so we're going to hire what kind of people to fit the job, right? So when we interview people, we have all kinds of people coming in, come to the FDA. Remember, FDA has a very good name. People wanted to come to work for that, for benefits, for everything, for the name, or whatever, they're going to come. So I have seen new pharmacy graduates coming in, applying for jobs. They say, well, I said, what do you want to come here? They said, well, we were told that FDA is a good place to work and have good benefits, good salary. Why not? I said, well, what can you contribute, right? You can't just come in as a new graduate and we have to teach you from scratch. everything, right? And what do I get from you? I was a little bit selfish. So I said, the best is for you. My best candidate for the work is a pharmacist with 5 years clinical experience. The best, if you have gone through the residency training and some study, publication, you know how to write, that's what I'm looking for. So now you just graduate, okay? So I think maybe you should go back and invest a little bit more money to go to another residency to become a clinical pharmacist, or you get another degree. Dr. Hoke mentioned about these days, the most secondary degree that people get is what? Remember? MBA. And the next one is MPH, right? So you can get a secondary degree to help you to get where you are, to be, most importantly, competitive with others. You get job satisfaction. So when I hire people like that, and it's the right fit, then there's very little turnover rate. This is very important for the managers, right? You can't deal with the people just coming and leave, like that. So it's the right people, and to me, clinical pharmacists is the right people I have hired all the time. Even now, I still have people working there. They haven't left yet. So they like the job, it's very rewarding. And of course, the benefit, the money, it's not bad. So the regulatory actions, after you have done the case evaluation, risk assessment, the agency can do the labeling change in the ADR sections, precautions and warning, including box warning for appropriate prescribing and informed patient use. Also, the company, if needed, they will be asked to conduct further post-marketing studies to understand the risk in the post-marketing setting. So this is very important as well. Product recalls, when the quality has problem, and the market withdrawal, when definitely the product has higher than acceptable risk, then just take it off the market. It happened many times in the past. And safety communication is very important. You, an agency or the drug company, should issue that to the public to prevent unintended consequences. So other safety-related pharmacovigilance activities include product quality, emergency-related outbreaks, and then also there's a counterterrorism scenarios you have to deal with, collaboration with other regulatory agencies, liaison with like a CDC and a health organization, physicians organization. So pharmacists' expanding roles in drug safety is really right now, it's very important to bridge to the future. What can we do? In clinical pharmacy setting, you can do detecting ADRs at the point of care. At the time you give the prescription out, medication out. And also, in the community pharmacy, you can educate patients and monitor the outcome about any side effects they have experienced, could have experienced, and then tell them how to manage it. If you find out you have skin color change, and the eye color change, yellowish, maybe they have an abdominal pain, maybe you have some liver injury, go to the doctor and stop the drug to see what happened. And then if you go to the industry, you may go to the pharmacovigilance department and safety data, running safety data analyst jobs there. You're going to be very, very good at that. And they are really requiring a lot of clinical pharmacists or even pharmacists doing this job. And research development, if you want to use, you want to have some dream, you want to develop a new drug in the discovery, then you can try to integrate the safety early in clinical trials to make sure you understand the safety and the risk and then manage it in early stage when you continue doing clinical trials without major problems. So actionable steps on patient safety. So I think in these 2 days, we have mentioned enough about patient-centered practice, and then as a point of care person, you need to stay informed and follow safety updates instead of falling behind. And then you can can advocate for digital or AI tools to improve safety tracking. I'm sure all the hospitals and industry, you have your own way to track this system about safety signals, safety information by drugs, by events, and then by labeling. If you want to, you can get trained in pharmacovigilance system and then get into the regulatory science, which is very, very hot these days. The company wants to use you to continue the development with the right regulatory support. You don't want to go in there blind and didn't know what's going on. Finally, I want to say something about the key to success for your profession is skills of communications. You have to think about how you can get the message across and convincing as well. So when you talk, you read, you have done enough reading, writing, and speaking, but can you convey the message across and convincing people listening to you, suggestions, proposal, recommendations? Think about that, right? You got to be solid to in your professional skills to support yourself, right? And then you also need to know how to convey the message. And here I had to mention that personality and authenticity matter, that not everyone can do this communication successfully. So people need to go out and walk out, talk to people, and look at people when you talk. All these things really just trying to make people have comfort in you and listen to you and take your recommendations. So, and then also you do know that you respect others. And finally, the most important thing I like is humors. When you convey the message with humors, people listen. Right? Okay, so let's hope that we can all reimagine the future by being a good pharmacist. Thank you.
Okay, thanks, Dr. Chen. So touching speech. So now we open several questions for the audience. Yes, please.
Thank you very much. Really wonderful, excellent, and a very vivid talk.
Thank you.
I think the central mainstay developing the drug has to be safe and effective. I'm particularly interested in one slide you presented that is related to the thalidomide,
yes,
Vioxx,
yes,
and also one the COVID-19 vaccine.
Yes.
Could you go back to that the
Sure.
slide?
Yeah, here, this
Oh,
one.
I'm particularly interested about the COVID-19 vaccine because I was deeply involved in the development of the COVID-19 vaccine. Even after this time, there's still a tremendous controversy about the COVID-19 vaccine. It is really safe and effective. Okay, the question is that the new NIH boss, Dr. Kennedy, you know, there's the CDC, the boss was hired, but just after 1 month was fired. And the CDC, many of the CDC staff, maybe close to 1,000, is going to be eliminated, and more is to be like, so there's a bit chaotic. Really, the problem is that Donald Trump's administration don't trust
Science.
Science, the mRNA vaccine. And they eliminate the funding, about $500 million in the funding. They are not going to fund the mRNA vaccine research. So many of the postdoc or the researchers are going to lose the job. So the chaotic. So the main stage that the question about the safe and effective or not, and still controversy, particularly myocarditis days, excess days, you name it, and many of them, the autoimmune disease. So, and Donald Trump even said, I want the data from the Pfizer or even the Moderna. Please disclose the safety data to the public, not to the White House, the president or the high-ranking officer. People deserve the right to know us about it, even at this time. COVID vaccine, the mRNA vaccine is only in the EUA authorized, not a BLA authorized. So in particular, the distribution data. So after vaccination, where the doctors injected through the mouth, where did they go? They go everywhere in the body. So that is where the problem. Even Pfizer, Moderna never distributed or disclosed the tissue distribution data. That is the most important. So my question is,
yes,
because that sentence is a little bit conservative. Does not disclose many of much of the information, just like a Vioxx thalidomide. Is the mRNA vaccine at this point still safe or effective or not? Your opinion, that's I'm particularly interested
okay.
because,
yeah.
Thank you very much.
Yeah. I believe in science, and I was really proud of being in the FDA because that agency is really science-based agency. They have hundreds of medical doctors in different specialty and hundreds of PhDs in different science, chemists, microbiologists, PK people, all PhDs altogether, thousands of people there. Okay, and my experience within the FDA is because I was a director, so I also attended high-level meetings most of the time to understand all the problems, issues in drug approval. Okay, so I know FDA most of the time respect each other's opinion. If this office and that scientist have different opinions, they will talk it out until you reach agreement. So every approval, the decision making or not approval, all science-based. You don't have a lot of discrepancy to the counter their decision. So almost every decision is like that. I'm not saying that there were no exceptions. For COVID, okay, it never happened before with such a quick turnaround from clinical trial all the way to patient use. Okay, and I don't know. Do we have time to tell you a very 2-minute story about COVID? At that time, what happened was President Trump actually was the one gathered a meeting, a roundtable meeting of all the manufacturers, companies that, including NIH, that NIH knows all the grants given to a different messenger RNA development lab or company, right? So he asked all the companies or the labs coming to the roundtable to discuss how are we going to develop the COVID vaccine right now, which I think we have a lockdown in April or March 2020. In May, they had a roundtable meeting, and Pfizer, Merck, name it, all there in the roundtable. And so Donald Trump said, who can actually develop the COVID vaccine fast enough to meet our needs? Pfizer said, oh, at least 1 to 2 years. Okay, and then you ask every other, it's all sometime. You know what? Moderna, do you know Moderna? Said 6 to 8 months. Donald Trump said, it's yours. They gave you all the money to develop quickly for the messenger RNA product vaccine. How did Pfizer get into the picture? Pfizer actually got the product from BioNTech in Germany, right? So they didn't have their product. They got their product from BioNTech. So eventually, these two products are being developed. Okay, both products about the same speed with almost 1 million people exposure worldwide. All different countries, different species, and different gender, and different doses. With that result, I'm just personal interest. I reviewed the data at that time, posted on the web. It's, I think, around 90% efficacy. So what are you going to believe? Are you going to believe that? Or you're not going to believe that? Say it's not effective. So are you going to take that? Yeah. You have no other options. So to move forward right now, do you think it's effective? It's good enough without causing any other problems? At that time, the evidence said it was effective. And the side effects, we don't know that much until you have opened it up to every country to get vaccinated. You collect the data, Pfizer, Moderna, they collect the data. So there are a certain percentage of the people in the United States, they don't believe in vaccine. They don't believe in COVID vaccine. So including Trump. But Trump got COVID, right? And then he got special treatment. So it's interesting in the sense at that time, you have no other options. You have to take the risk. And then you know what? It was working, right? It's the infection rate. Right? The rate decreased. So let's go to my area. It's called adverse event profile. You go to the website. There is called the FDA FAERS, F-E-R-S, database. You go in there. There is a public website. You can get all the data you want for any drugs, including the vaccine. You go in and take a look and see, can you analyze it? It's not easy. It's not easy. When you have such big exposure, the number just jumped 10 times or 100 times. It's not easy. So does that mean it's causing the problems? That's called, I think that girls I talked to him yesterday from his study. In the end, you do all this study, it's really useful. So I call that, is it art or science? Sometimes it's really not science, it's art. You have to make a decision based on other factors. Okay, the best is to have the science supporting it. So the adverse reactions area or safety area is really, really difficult. If you experience that, forget it. It's devastating. Just no way I can believe the drug is safe, right? But in the meantime, there's so many people benefited from it. What are you going to say? The drug is not good enough? So it really depends. Risk and benefit balance. What are you going to do then? Okay. So let me just give you one more final thoughts on the COVID vaccine. Right now, it's how many years past forward, right? So they are still developing COVID vaccine. Do you want to take it? I got shots 5 times, okay? It's all free anyway. So I had the shots 5 times. Every time a little bit reaction, that's fine. But you know what? I still got COVID. Infected from my grandchild, right? So it's not full protection at all. However, do you want to take it? It's your decision. But what I believe personally is that it's all related to your immune system, protecting yourself or not. Yeah, if you can eat well, sleep well, exercise, you have perfect immune system to fight anything, including cancer. This is just ideal situation, you know? So nowadays, COVID-19 is not that damaging at all, because the strength is just keep weakening. Do you want to take more vaccine, COVID-19 vaccine? It's up to you. When I come here, before I came here, people ask me, you better take another shot of vaccine. I said, no, I believe in myself that I can take care of myself, that I don't need that. I don't need that. So it's up to you how you're going to do it. You say, well, there's so much side effects. I don't believe it. Injury in your system. People have like arthritis for no reason, and a heart problem for no reason. There will be rare scenarios in different kind of people when the drug is exposed to such a big, wide population. So you have to take that seriously to make a decision, and then once you feel something wrong with the medications or vaccine you had, go where? Go doctors. Get treatment right away. Don't get yourself into the situation that actually get yourself in the, you can't come back anymore. So that's the message I want to get across about drug safety. Don't be afraid. You're going to take advantage of it, but you're going to also take care of the risk yourself with doctor's help. Is that okay?
Okay. Okay, for the time, thanks, Dr. Chen, because it's more challenge for the pharmacist with the continuously advanced drug development. So it's getting more important. Okay, thanks for Dr. Chen's speech.
Yes, thank you, Dr. Chen, for answering that question. And we do have gifts for you, so if you could stay on the stage as our moderator provides you with the gifts. All right, thank you very much. Well, we reached the conclusion of the conference, and I would like to extend my sincere gratitude to all the distinguished speakers, guests, and participants who joined us over the last 2 days. Your expertise, ideas, and enthusiasm have made this conference a true success. And a special thanks to the organizing team for their dedication and support in bringing this international event to life. Now, to officially conclude the 2025 International Pharmacy Conference, we would like to invite Dean Weichao Zhang to give their closing remarks. Dean Zhang, the floor is yours.
Okay, many thanks for Professor Lee for your wonderful talk. Without art, science is spiritless. But without science, art is rootless. As we come to the close of the conference, first of all, I would like to express my gratitude to Dr. Ellen Zhao for his tremendous support. Without his support, this conference would not have been able to possible. Second, I would like to thank to the organizing committee led by Chen Xiangyin 主任、美泉老师、雅慧老师、玉慧。Thank you very much for your dedication and hard working. And of course, to all the speakers, your contributions have enriched the meeting and create valuable opportunities for knowledge exchange and international collaborations. The 2 days conference focused on pharmacy education, clinical pharmacy, genetic testing, counseling, AI-driven drug discovery, as well as pharmacovigilance. The future of pharmacy will depend on our ability based on pharmacy professionals to bridge pharmaceutical science and clinical practice. with the vision, with the commitment shown today, I'm confident that we are prepared to facing the change, to facing the challenge ahead. Finally, I wish you safe travel, continuous success. Enjoy the time in Taipei. I'm looking forward to seeing you next conference. Enjoy the time and have a nice day. Thank you so much. Thank you.
Thank you, Dean Zhang, for the closing remarks. And yes, that officially concludes our conference. So thank you so much for your participation and attendance. We'll hope to see you again next year. Thank you.